Okay everyone, thank you for being on time, those of you in the private Zoom webinar room. I see that we have, let's see here, 19 out of the 22 students who registered for this webinar are in attendance and on time. Again, thank you for being on time. So this is going to be a revision webinar where we're going to be focusing on the subject of energy in biological systems. Once again, this is going to be a webinar which largely is intended to help students who are studying for the AQA new spec biology A-level specification. However, the topics that we're going to be covering in this webinar are equally applicable to those of you studying under OCR and Edexcel. I'm also expecting, let's see here, 6 students from Acklam-Burley and the Le Swap's consortium of schools to be joining us, but as yet they have not logged in, and by prior arrangement, as with the previous webinars, only the students from the Le Swap consortium, only their chat will be visible on the screen at any given time, so I'll bring that up on the screen as soon as they've logged in. Now, just to quickly have a look at what we're going to be going over in this webinar, let me just share my screen with the Zoom viewers as well. So, as I say, we're going to be covering this topic. This is actually the A-level specification from AQA. Just so I can show you, it's this document here, which you can download from the AQA website. Now, those of you studying under OCR and Edexcel, you will have an equivalent section in your specification where bioenergetics, things like respiration and photosynthesis are covered. So I know that we have a few of the students from Invisible College who are in the private Zoom room who are studying under OCR and Edexcel, so I would encourage you guys as well to have this specification document or your equivalent specification open while we go through this topic. Now, with the A-level topics that we're going through in these webinars, there's an awful lot of detail, and we've only got 3 hours to cover it. So as with the previous webinars, if it turns out that we run out of time or you guys are unwilling to continue beyond the 3-hour mark, I will schedule an additional webinar to cover any topics that we don't manage to cover in this revision webinar here. So the topic for today's webinar is energy transfers in and between organisms, which is topic 3.5 in the AQA specification. We're going to start by looking at the biochemistry of photosynthesis. We're going to move on to looking at the biochemistry of respiration, followed by the importance and the significance of energy and energy transfers in the ecological context. In other words, looking at the ways in which energy is transferred between organisms and also between organisms in their environment. And we're going to finish off by looking at nutrient cycles, focusing particularly on the nitrogen and phosphorus cycles. So those are the topics that we're going to be covering. Now, as I say, this is a revision webinar, and while I do try and go into enough depth to kind of cover the concepts and the ideas and the processes in as much detail as possible, obviously we are somewhat restricted by the fact that we have only 3 hours to do so. So those of you who are already students and at my website and are enrolled on some of the course modules, the students at the Le Swap consortium and also many of the private students already have access to these course modules. Now, if you are interested in some more in-depth teaching on this particular topic, you'll find that in this course module over here, Energy and Biological Processes, and in this course module there are a number of lessons on each of the topics that we're going to be covering, and each of these will have a in-depth teaching video where we go into these topics in a lot more depth. Now, I've left out a couple of these things here. So for example, we're not going to be going into the experimental evidence for photosynthesis, and we're not going to go into too much depth about the different kinds of autotrophs and heterotrophs, but if you are interested in that, then those of you who are already enrolled, you can find that information in this course module here. So let's get started then. So let's start with the biochemistry of photosynthesis. So we'll begin with looking at the light-dependent reactions. Now, the site where these biochemical reactions and pathways take place is the chloroplast. And let me just make sure that my screen is actually being shared here with the Zoom attendees, because I can see a few of you saying that you can't see my screen here. Sorry about that. Hopefully you guys can see that now. Great. Okay, so as always, as we go through this webinar as well, please do type any questions that you may have in the Q&A box and not in the chat box, because again, the Zoom chat tends to scroll by quite quickly and I may miss your question, whereas if you put it into the Q&A box, I will address any questions that you guys ask at the end of this webinar when we turn the YouTube live stream off. So the chloroplast is the site of the light-dependent reactions. Now hopefully in your revision of the topic of cell biology, you will have gone into the ultrastructure of the chloroplast in some depth, but we'll quickly make reference to it here. Now, the actual biochemical reactions which are involved in the light-dependent pathway, these take place within the thylakoid membranes of the grana. So this is one granum here, and you can see here that we see the thylakoid membranes of the thylakoids which are stacked into that granum. So these reactions begin when the energy in sunlight is captured by the pigments located within the grana. So we're going to be looking at some of these pigments. Now, it's worthwhile mentioning here that we need to kind of make reference to a little bit of physics here. So light energy can be thought to be packaged into quanta called photons, or packets called photons. Now, photons essentially are a way of understanding light in terms of light being composed of particles with specific energies. So the energy content of one of these packets or one of these individual quanta of light depends upon the wavelength of the light. Now the reason for this is because the energy carried by any kind of electromagnetic wave is given by the equation E is equal to h multiplied by f. So E is equal to a constant, Planck's constant, multiplied by the frequency of the wave. But light can be thought of as waves or as particles. So when we think about it as photons, we're making reference to an idea from quantum mechanics and quantum theory, where energy is quantized and not continuous at the level of objects as small as a photon. So the energy content of a photon depends upon the wavelength or the color of the light. In general, the shorter the wavelength, the higher the energy carried by that photon. Now, the energy associated with the photon of light, as we say, is called a quantum of energy. Now, this comes from the Latin quantum, which means how much. And the idea of quanta is basically the fundamental idea which gives rise to the science of quantum theory, where in the macroscopic world, we're familiar with things like energy and length and mass and things like that as being continuous quantities. In other words, you can think of energy as being a continuous variable which can take any numerical value on the macroscopic scale. And that idea works on the macroscopic scale. But on the submicroscopic scale, when you're talking about things like electrons and photons of light, it turns out that we can more usefully think of things like light energy as being packaged into discrete quanta. So you can have either one level of energy or the next level of energy, but nothing in between. So this comes from quantum theory, and the fundamental idea there is that things like light are packaged into discrete units that we call quanta. So blue light has a relatively short wavelength, whereas red light has a relatively long wavelength, and consequently blue light carries the higher energy. Now, plants are generally able to absorb a very wide range of wavelengths because they contain a different variety of pigments or coloured chemicals with different structures and absorption properties. So we're going to be looking at that, but in order to do that, let's just quickly make reference to a couple of basic ideas here. Now, visible light represents only a very small fraction of the full spectrum of frequencies and wavelengths of electromagnetic radiation which is produced by the sun. So we find that the sun produces a wide range of electromagnetic waves, including all of these that you see here, which make up the electromagnetic spectrum. Now, within that spectrum, within that range of wavelengths, visible light makes up a relatively short or a relatively narrow range of wavelengths. So we can see those wavelengths displayed here, all the way from around about 400 or just under 400 nanometers, all the way up to about 750 nanometers. So here we're going from the shortest wavelengths, the blue and the violet, up to the longest wavelengths, the red. So plant pigments will absorb light energy with different wavelengths that fall within this visible spectrum. So let's look at absorption spectra then. So in general, And again, we're kind of looking at fundamental ideas here to build up a better understanding. But when a beam of light, a beam of white light, is passed through a glass prism, the visible spectrum is obtained by the dispersion of that white light into its component frequencies and wavelengths. Now, this was discovered by Isaac Newton. And so we take a beam of white light and we find that when it's dispersed by refraction through a prism, we find that the different wavelengths of light are split and they can be projected onto a screen to display all of the different visible colours of light that make up white light. Now we can do a similar thing. We take a white beam of light and pass it through a solution of a leaf extract. So what we've done here is we've taken a leaf, we've crushed it up, and we've extracted the pigments from that leaf and produced a solution of those pigments in water, and then we pass a beam of white light through it. Now what we find is when we then project the light which is transmitted through that solution of the plant extract and then disperse it in a prism, we find that there are certain wavelengths missing. In other words, we find certain wavelengths are transmitted, like this blue wavelength here and this range of wavelengths here between the green and the yellow, this band of yellow wavelengths here and this band here, but we find that there's conspicuous absences of other ranges of wavelengths, and this tells us that the materials in that solution have absorbed these ranges of wavelengths. So these black bands appear in the spectrum where particular wavelengths of light have been absorbed by the plant extract. So predominantly we find that it's the wavelengths of the blue and the red ends of the spectrum which have been absorbed by that light. Let me just make myself a little bit smaller here on the screen so you guys can see that text a little bit better. There we go. Let's just move that over here. A little bit of ninja editing on the fly here, sorry guys. Just trying to make things a little bit more visible for you. There we go. Okay, so let's have a look at the implications of this for studying the ways in which plants absorb various wavelengths of sunlight energy. So this spectrum that we've just produced is referred to as an absorption spectrum. So we passed a beam of white light through a leaf pigment, and then we dispersed the transmitted light from the light that had passed through that leaf extract through a prism, and we've produced this absorption spectrum which tells us which wavelengths and which ranges of wavelengths the pigments from that plant have absorbed. Now we can get more detailed information about the absorption process by using a device called a spectrometer. Now the degree of absorption at each of these wavelengths can be measured by for either the total plant extract, or if we choose to separate the pigments that make up that plant extract, by the individual pigments. So this information can be used to generate what we call an absorption spectrum graph. So in a similar way, the effectiveness of different wavelengths of light in bringing about photosynthesis can also be experimentally measured. So the rate of photosynthesis at different wavelengths can be then measured so that we can find which wavelengths are responsible, or in other words, which wavelengths of light are the ones that are absorbed by the plant in order to capture the energy carried by those electromagnetic waves and use it for photosynthesis. Now, the rate of photosynthesis at different wavelengths, when we experimentally measure that and represent it as a graph, we refer to that as an action spectrum graph. So let's have a look at the relationship between absorption and action spectra. So what we have here is the absorption spectrum for a particular species of plant, and we see here that the percentage absorption, so in other words the percentage of the incident light intensity which has been absorbed at these wavelengths is shown by this curve here. Now the action spectrum is shown by this other curve here. Now although all wavelengths between about 400 and about 700 nanometers are absorbed by the leaf extract to some extent, as we can see here, the maximum absorption occurs in the blue-violet end of the spectrum and in the end of the spectrum nearest the red, as we can see here. So we see here that there is actually quite a close correlation between the action spectrum of the plant and the absorption spectrum of the plant. So in other words, the rate of photosynthesis is largely determined by the light of different wavelengths being absorbed. Now, as we can see here, the green wavelengths in the middle of the spectrum is generally not absorbed anywhere near as much as the blue and the red wavelengths. In fact, these wavelengths are largely reflected from the plant, and this is the reason why most plants appear green. It's because they are reflecting the green wavelengths of light back towards the observer, whereas they are absorbing the purple and violet and blue wavelengths and the red wavelengths at the other end. So, pigments can be extracted, as we said before, by finely chopping and grinding the plant tissues, particularly the leaves, and we do this in a solvent called acetone. And acetone is an extremely good solvent for a number of different applications, both in laboratory and in other applications. So, when the pigment extract is subjected to ascending paper chromatography, the pigments can then be analysed and identified using this method. So, many of you will have conducted this type of experiment for yourselves. So we can see here the origin line where the original pigment mixture dot was placed, and the solvent front has moved up this chromatography paper and has separated the components of that pigment mixture, and we can then go on to identify these pigments by further analysis. And these are the identities of these photosynthetic pigments. Now, the principal plant pigments found in green plants are the chlorophylls, both A and B, and also the carotenoids, which include carotene and xanthophyll. Now, the role of these pigments, as we said before, is essentially to capture the packets of energy, the quanta of light energy, and each of the pigments will absorb various wavelengths of light to different extents. Now, the reason that they appear these different colours is because each pigment will reflect particular frequencies which it does not absorb. So we see here that chlorophyll B is reflecting green frequencies, chlorophyll A tends to reflect the kind of blue-green frequencies here, and so on. So, at advanced level, you need to have some understanding of how these photosynthetic pigments are organised in the plants and in the cells of the plant. Now, the photosynthetic pigments tend to be embedded, as we said before, in the thylakoid membranes of the chloroplasts, of the grana of the chloroplasts, and they are embedded in clusters which are called photosystems. Now, I'm going to use this diagram here to represent the arrangement of these pigment molecules in such a photosystem. So each photosystem consists of specialized chlorophyll molecules, and one of these is called the reaction center, and these are surrounded by a few hundred pigment molecules that make up what we call the antenna complex, which you see here. And the reaction center is what we see down here. Now, I've kind of arranged them in this diagram, not necessarily because they're arranged in this way in the actual biochemical context. However, you can see here that the diagram is built around a funnel shape. Now, the reason why we've kind of used this analogy of a funnel should become clear shortly. So, the antenna pigments will gather the light energy of various wavelengths and funnel this energy towards the reaction center. So, the incoming light energy is absorbed, and then the electrons are passed, as we'll see, between these various pigments and molecules, until eventually the energy that was originally carried by those photons of light energy is passed to this specialized chlorophyll molecule that we call a reaction center. So in this way, the chlorophyll molecule at the reaction center eventually receives all of the absorbed energy which have been absorbed by all of these various pigment molecules which make up the antenna complex. So let's look at the machinery of the light-dependent reaction. So the thylakoid membranes of the grana contain two types of photosystems. Now, we refer to these as photosystem 1 and photosystem 2 respectively. Now, before we go any further, through the course of this webinar, we're going to be making reference to a number of diagrams very similar to this for both when we revise the biochemistry of photosynthesis as well as the biochemistry of respiration. Now, on the vertical axes of these diagrams, we have the redox potential. Now, this is perhaps a term which it may be unfamiliar to some of you who don't study advanced level chemistry, but to those of you who don't study advanced level chemistry, you can simply think of this axis as representing an energy level for electrons. So we go from the lowest energy level at the bottom of this axis to the higher energy levels at the top of this axis. So the positions of these two photosystems, again, each of these photosystems is made up of an antenna complex and a reaction center. The relative positions of these two photosystems on this diagram represent their relative energy levels, or the energy levels of the electrons located in these photosystems. So we're going to be making reference to that as we go on. So the two photosystems differ because of the wavelength of light that the reaction centers of these molecules absorb. Now these the positions of these two represent what we call the redox potential, but we can think of this simply as the energy level or the energy content of the electrons involved in those systems. So we see here that relative to photosystem 2, photosystem 1, or the electrons in photosystem 1, are at a higher relative energy level than those of photosystem 2, and conversely the electrons involved in photosystem 2 are at a relatively lower energy level than those involved in photosystem 1. So we can think of this axis simply as representing the energy level of the electrons. So bear that in mind as we go through this explanation. So also we find within the thylakoid membranes of the grana, we find a number of other molecules and a number of other bits of biochemical machinery, if you like. We firstly find molecules called primary electron acceptor molecules, and again, in terms of their relative energy levels, we find these at these locations relative to the photosystems and each other. We also find what we call carrier molecules, which form part of something referred to as an electron transport chain. Now, the concept of an electron transport chain, hopefully you'll remember, is also relevant and also important in respiration as well as photosynthesis. So here is the electron transport chain. So these are molecules whose function is simply to pass electrons along from one to the other. And you see here that in terms of relative energy levels, this one is at a higher energy level than this one is at a higher energy level than this one as well. So we also find another molecule, which is an electron acceptor, which is called ferredoxin, which is at this energy level here. So all of these different molecular components or parts of the machinery, if you like. Now I'm using the word machinery in a kind of euphemistic or a kind of as an analogy, because really we're talking about living systems and not machines. However, because of the way that these molecules function, you can think of them as parts of a very complicated biochemical machine. So when we use the term machinery here, it is kind of a euphemism somewhat. So all of these components are required for the light-dependent reactions of photosynthesis, and these are ordered on this chart to represent their relative electron energy levels, or their redox potentials. So let's have a look at what happens and how all of these different components of the light-dependent reactions are involved in capturing the light energy involved in photosynthesis. So firstly, when this light strikes the grana of the chloroplasts, the antenna complex molecules, the pigments within those antenna complexes, will absorb the light energy. Now, by absorbing light energy, the electrons within those pigment molecules enter what we would refer to as an excited state. Now this is kind of a concept which you guys have probably been learning about from, I would say, probably year 9 onwards. The idea that electrons exist within atoms at discrete energy levels, and in order for an electron to jump from a lower to a higher energy level, it needs to absorb enough energy in the form of an electromagnetic wave or a photon in order to promote it from that lower energy level to a higher energy level. And that's in fact what's happening within the pigment molecules found in these photosystems when they absorb the electrons within these pigments absorb photons. So energy is funneled, as we saw before, to each of the reaction centers, where the chlorophyll molecule in that reaction center receives sufficient energy to release an electron. Now, when an atom within a molecule releases an electron, we would say that that atom, and generally the molecule as a whole, has become ionized. So when this is caused by the absorption of a photon, we would refer to this as photoionization. So a single electron is released for each photon which is absorbed, Now, because each of these photosystems needs to absorb 2 photons in order to get this process underway, this means that in each of these photosystems, once the energy from those 2 photons has been funneled down to the reaction center will release 2 electrons for each one of these photosystems. So the released electrons are accepted by the primary acceptor molecules located again within those thylakoid membranes. So again, this is happening in both photosystems here, and we see that they are both at different relative energy levels, and so ultimately the electrons which are passed to these primary acceptor molecules are also at different energy levels. So the release of the electrons from the photosystems creates what we can refer to as electron holes at the reaction centers. Now these must be filled if photoionization is to occur again. So the electron hole in photosystem 1 is filled because the electrons pass down the chain of carriers from the primary acceptor for photosystem 2 in this manner. And so these 2 electrons, which were promoted to the primary acceptor by the photoionization of the reaction center in photosystem 2, ultimately get funneled down the electron transport chain by these carrier molecules from the primary acceptor back to the reaction center of photosystem 1, and this fills the so-called electron hole left by the promotion of these 2 electrons by the photoionization of this reaction center here. So this flow of electrons along this chain, because each of these carrier molecules is at a successively lower energy level, the energy difference has to go somewhere because of course energy is conserved. So in fact this energy, the energy difference between these carriers at each one of these steps is used to form ATP from ADP and inorganic phosphate, as we'll see later on. So the formation of ATP as the photoexcited electrons pass along this chain, this is referred to as photophosphorylation, because what we're doing here is we are phosphorylating ADP to form ATP, but we are doing so by making use of the energy released by the movement of electrons from a carrier molecule at a higher energy level to a carrier molecule at a lower energy level. And because this is all ultimately powered by energy carried by light, this is referred to as photophosphorylation, or phosphorylation using light. So we've explained what happens to fill the electron hole at photosystem 1, but of course we still have this electron hole left in the reaction center in photosystem 2. Now this is filled because ultimately water molecules will donate electrons to the chlorophyll molecule at the reaction center. Now again, we start with a water molecule at this relative energy level, or redox potential, and the splitting of water to form oxygen and hydrogen ions is ultimately accompanied by the release of 2 electrons, and these 2 electrons are then used to fill that hole in the reaction center in photosystem 2. Now the donation of the electrons to photosystem 2 is what results in the splitting of the water molecule, and this releases again hydrogen ions and oxygen gas, and it's actually this is the origin of the oxygen gas released during photosynthesis. So ultimately we can think of oxygen gas as a waste product of photosynthesis. Now this chemical reaction here is referred to as photolysis of water, and again it is lysis meaning breaking apart and photo meaning to do with light. So this literally translates as splitting with light. So water is being split by using light as a source of energy. So now the electron holes of both photosystems have been filled and the whole process can begin again. So we can make use of this biochemical mechanism in the chemiosmotic theory. Now basically what this says is that as the light energy carried by those 2 photons are received by photosystem 2, a pair of electrons are excited to a higher energy level. Now these electrons, as we said before, travel down the electron transport chain, and this is ultimately what causes the hydrogen ions to diffuse across the thylakoid membranes and into the inner thylakoid space. Now Once they're in the interthylakoid space, the hydrogen ions are then transported down their concentration gradient through an enzyme called ATP synthase, which is actually found embedded within the thylakoid membranes themselves, and this creates ATP by the phosphorylation of ADP to ATP. Now, this is very similar to the idea of the chemiosmotic hypothesis behind the powering of the reactions across the membranes of mitochondria, as we'll see when we look at respiration. So the movement of ions, in this case the protons, across a selectively permeable membrane down their electrochemical gradient, this is called chemiosmosis. Now the reason is because osmosis, just the word osmosis on its own, refers specifically to the movement of water molecules down their concentration gradient across a selectively permeable membrane. But here we're not talking about the movement of water molecules, we're talking about the movement of hydrogen ions, and so this is termed chemiosmosis. So chemiosmosis is essentially is essential to the generation of ATP in this context, but also in the context of cellular respiration as well. Now this derives from the fact that hydrogen ions diffuse in a way which is similar to how water molecules diffuse in osmosis. So photophosphorylation is the production of ATP using the energy of sunlight, and photophosphorylation is made possible as a result of chemiosmosis. So, the electrons released from photosystem 1 to the primary acceptor molecules are then passed to the ferredoxin molecule. Now, this happens ultimately so that the ferredoxin molecule can go on to pass those two electrons down to the molecule NADP+, which is a hydrogen carrier. And again, NADP is nicotinamide adenine dinucleotide phosphate. So we see here that the primary acceptor molecule is at this redox potential or energy level. It then passes those two electrons down to the ferredoxin molecule, which is at a relatively lower energy level, and then the ferredoxin molecule goes on to pass those two electrons to the hydrogen carrier NADP. So these electrons, together with the hydrogen ions formed during the photolysis of water that we mentioned before, is what are responsible for reducing NADP to NADPH. Now you have to be very careful with the words oxidation and reduction, because at advanced level, even if you're not studying advanced level chemistry, it's important that you remember that the words oxidation and reduction, both in biochemistry and in just chemistry, can have three different definitions. So oxidation and reduction can refer to the addition and loss of oxygen atoms from a molecule respectively, or they can also, oxidation and reduction can also refer to the loss of electrons and the gain of electrons by an atom or a molecule respectively, but also oxidation and reduction can refer to the loss of hydrogen ions or hydrogen atoms from a molecule, or the addition of hydrogen atoms to a molecule respectively as well. And based on the context, you need to know which is being referred to. So in this case, when we talk about the reduction of NADP+, we are referring to the fact that the electrons, together with the hydrogen ions formed here, are being used to add hydrogen to the NADP+, and therefore this matches the description of or matches the definition of reduction with respect to the addition of hydrogen ions. So the hydrogen ions from here are being promoted to a higher energy level, and then eventually are being passed down to NADP along with the two electrons to form a more stable product at a lower energy level, NADPH, plus a free proton. So the light-dependent stage of photosynthesis has generated ATP and has also generated reducing power in the form of NADPH and protons. Again, reducing power can we'll see how that's later involved in the further production of ATP. So ATP and NADPH, or reduced NADP, these are both needed to drive the light-independent stages of photosynthesis. So let's quickly summarize the light-dependent reactions in terms of what we call the Z scheme. So we have the absorption of two photons by the two photosystems. This then causes the promotion of two electrons from the reaction centers of those photosystems to primary acceptor molecules at these relative redox potentials here. In so doing, we are also photolysing the water molecule to form oxygen, which is essentially a waste product which then diffuses out of the plant, and some of which is used in respiration, but also, more importantly, the production of hydrogen ions and also 2 electrons, which are then used to fill the so-called electron hole in photosystem 2. So the next step is the transfer down this electron transport chain of carrier molecules from the primary acceptor, which accepted the 2 electrons which were promoted from the reaction center of photosystem 2, down these successively lower energy leveled carrier molecules, eventually down to fill the electron hole in the reaction center of photosystem 1. And in so doing, these transfers of energy from these successively higher to lower energy levels mean that the differences in energy can be used to drive the phosphorylation of ADP to form ATP. So we refer to this as photophosphorylation. In turn, the primary acceptor, which accepts the 2 electrons which have been promoted to that energy level by the absorption of photons in photosystem 1, these are then passed down to the ferredoxin molecule, which then in turn passes them down to a lower energy level at the NADP molecule, and the 2 protons produced by the photolysis of a water molecule are then added along with the 2 electrons to that NADP to reduce it to form NADPH and a free proton. So the purpose of the light-dependent stage is to generate both ATP directly by photophosphorylation and also to generate reducing power in the form of NADPH for use in the light-independent stage of the reactions of photosynthesis. Now, I realise that I've gone through that in rather quickly, but again, those of you who want more detail on that, please do have a look at that course module on my website on the subject of bioenergetics for advanced level, because we go into that in a lot more depth with lots more examples as well. So we've looked at the light-dependent reactions. Let's have a look now at the light-independent reactions. Now, many of you will have, well, all of you hopefully will recall that at GCSE level, the only biochemistry of photosynthesis that you would have been made familiar with is the overall summary equation of to represent photosynthesis. Now, this is incredibly oversimplistic, as many of you have realized during the course of your A-level course. So the overall process of photosynthesis in eukaryotes can be expressed by that summary equation. However, this is an incredibly simplified representation. Firstly, photosynthesis does not occur in one chemical step. It occurs in multiple chemical steps. And we've seen that already in the light-independent light-dependent stages of photosynthesis. So this equation shows that overall, across both processes of the light-dependent stage and the light-independent stage, 6 molecules of carbon dioxide react with 6 molecules of water to form 1 molecule of glucose and 6 molecules of oxygen through the absorption and the utilization of light energy. However, this hides many of the intervening reactions, or the kind of intermediate stages of this process, and many of these are driven by ATP and also the reducing power of NADPH. So the precise biochemical reactions in which these 6 molecules of carbon dioxide are utilized are called the Calvin cycle. Now, we're not going to get into the experimental evidence for this, because again, we went into that in depth within the course module on my website, and this is really just a kind of revision session, and also because the AQA specification does not explicitly require you to know about the background of the experimental evidence, but we are going to look at the biochemistry, because that is something that you do need to know. So, Melvin Calvin and his co-workers determined the chemical steps involved in the reduction of carbon dioxide to carbohydrates during the light-independent stage of photosynthesis, and he was awarded the Nobel Prize in 1961 for this work. Now, the experimental procedure actually involved the use of radio-labeled carbon, or the radioactive carbon-14. So, by providing cultures of single-celled algae with radioactive carbon dioxide, he was able to follow what happened to that carbon dioxide through that series of biochemical steps, and essentially he used those experimental measurements and evidence to back up a scheme of biochemical reactions which explained the production of glucose from carbon dioxide. So let's look at the first stage, which is called CO2 fixation. So the light-independent stage of photosynthesis takes place not in the thylakoid membranes, but in the stroma of the chloroplast. Now, the first stage, as I say, of the Calvin cycle is referred to as carbon dioxide fixation. Now, located within the stroma of the chloroplast is the 5-carbon phosphorylated sugar known as ribulose bisphosphate, or RuBP for short. Now, we can visualize this in a kind of simplified manner by thinking of it as a 5-carbon sugar, the carbons represented here, and with 2 phosphate groups attached to the ends of that linear 5-carbon chain. Now the enzyme Rubisco, which is an abbreviation for ribulose 1,5-bisphosphate carboxylase or oxygenase, this catalyzes the reaction between the carbon dioxide and the RuBP, and this is what is referred to as carbon dioxide fixation. Now this reaction forms an unstable 6-carbon compound, and this immediately splits, because of its instability, into 2 more stable molecules of the compound glycerate 3-phosphate, or GP for short. Now again, we are not showing the full structural formulae here because they're not necessary for you guys, but we can again visualize these more usefully in this way here. So again, these are the carbon atoms within these 2 molecules of GP, and these are the 2 phosphate groups. So glycerate 3-phosphate is actually the first stable compound which is formed during the Calvin cycle. So let's look at the next stage, which is called, which we can refer to as the reduction of glycerate 3-phosphate. Now, this involves the reduction of these molecules to a 3-carbon sugar called, which we can refer to as a triose phosphate. So here we are representing the molecular formulae of these, just to kind of give you some clarity as to what's actually happening to these molecules here. So the second stage involves the reduction of these 2 glycerate 3-phosphate molecules to form triose phosphate molecules. Again, we have 2 of those molecules here, 3 carbon sugars, and this requires the hydrogen from NADPH, and so it's an endogonic or endothermic energy demanding reaction which absorbs energy. So this requires the use of energy from the hydrolysis of ATP molecules. So in order to hydrolyze, or rather in order to reduce 2 of these glycerate 3-phosphate molecules to form 2 triose phosphate molecules, we need the hydrolysis of 2 ATP molecules and the resulting release of energy to power this process, but we also need the reducing power of 2 NADPH molecules, which are oxidized to form 2 NADP plus molecules. So this also produces a water molecule. So the conversion of GP to TP utilizes the products of the light dependent stage. So again, we got this ATP from the light dependent stage. We also got NADPH from the light dependent stage as well. So the third stage is what we can refer to as ribulose regeneration. So here we have 2 triose phosphate molecules. Now, as the triose phosphate molecules accumulate, some of these, not all of them, but some of these are used to form glucose, which can then be used in respiration and converted into structural carbohydrates for the cell walls, storage carbohydrates such as starch, or they can also be used to manufacture other essential food molecules which the plant needs, such as amino acids and also fatty acids. But some of the triose phosphate molecules are used to recreate or regenerate the compound RuBP to enable the cycle to continue to operate. So this stage again utilizes ATP for the phosphorylation of RuBP, because here we've reformed this 5-carbon ribulose phosphate molecule, but we need to reform ribulose biphosphate. So we need to add a phosphate group, and we get this phosphate group from the hydrolysis of ATP, and we also require energy to power this process to form this molecule from this molecule, and again that energy comes from the hydrolysis of an ATP molecule. So let's summarize the Calvin cycle quickly here. So we have the carbon fixation stage, followed by the reduction stage. Now some of these triose phosphates again are used to produce various products which the plant needs, but some of them are used to reform ribulose bisphosphate, and again we can refer to this as the ribulose regeneration stage of the Calvin cycle. So overall, 6 molecules of CO2 will combine with 6 molecules of RuBP to form 6 molecules of an unstable 6-carbon compound that ultimately splits into 12 molecules of the 3-carbon compound GP. So 2 molecules of triose phosphate are used to form 1 glucose molecule in the product synthesis stage, and the remaining 10 molecules of the triose phosphate can then be used to regenerate 6 molecules of ribulose bisphosphate in this manner. And to do that, we would of course need 6 molecules of ATP to power that process, and also to provide the additional 6 phosphate groups to form those bisphosphate molecules. So, now, it all looks very complicated, but the reason why I summarized it first in terms of one set, so if I go back here, so I want you guys to try and memorize the, if you have to memorize one of these Calvin cycles, it's probably best, if you don't want to take the time to memorize all of this, to memorize this, because here we are talking about what happens when we feed 1 carbon dioxide molecule into this process. So these are the relative numbers of these intermediate products of the Calvin cycle which are involved when we take 1 carbon dioxide molecule. But in order to form 1 glucose molecule, this process needs 6 CO2 molecules. So you need to then multiply the relative or the individual intermediates in this process by 6 in order to get the actual number of molecules involved in producing 1 glucose molecule through this process of the Calvin cycle. So let's have a look at one more summary of this process just to really hammer it home. So 2 out of 12, or 1/6 of the triose phosphate molecules are used to form the 6-carbon single glucose molecule here. So 10 out of the 12, or 5/6 of the triose phosphate molecules that we start with, that we begin with here at this stage of the Calvin cycle, are used to regenerate 6 molecules of RuBP. So here we're talking about 30 carbon atoms involved overall. So the importance of the light-dependent stage for the Calvin cycle, again, is because through the course of his experiments, Calvin actually measured the relative amounts of glycerate phosphate and RuBP present first in a continuous light, but then when the light was switched off. Now, I'm not going to go into, again, too much of the detail as to the background of Calvin's experiments, but you do need to understand some of the kind of experimental basis for this, because I've seen a lot of exam questions both in recent years and previous years which will present you with this information, and examiners will typically do that. They will present you with information from an experimental context which you may be unfamiliar with, and they'll require you to apply your understanding to that unfamiliar context in order to interpret those experimental results. So he measured the relative amounts of these two substances, firstly under conditions where there was continuous light, and then under conditions where the light was switched off. Now in the light, both GP and RuBP will remain at a steady level, but in the light, RuBP is converted into GP, as we saw before, which in turn is converted back to RuBP during the ribulose regeneration stage. But in darkness, the GP will accumulate and the levels of RuBP will fall. Now this again provides evidence for the fact that the Calvin cycle becomes disrupted in complete darkness, and this effect is observed because the reduction of GP to TP again requires ATP and NADPH, which were formed from the light-dependent stage. So the formation of the triose phosphate is therefore prevented, and the regeneration of RuBP cannot take place because the products from the light-dependent stage which are required for those processes are not present in sufficient quantity. So what we're saying here ultimately is that the Calvin cycle is dependent upon the light-dependent stage for its operation, even though the Calvin cycle is what we will refer to as part of the light-independent stage. So we can summarize photosynthesis overall by saying that photoautotrophs which make use of photosynthesis will do so by essentially undergoing a two-step biochemical process. During the light-dependent stage, it generates ATP and NADPH, and then utilizes those products to reduce carbon dioxide to carbohydrates in the light independent stage. So the two stages, even though they occur under different light conditions, are linked to each other. They are dependent upon each other, as we can see here in this diagram. Now, you do need to know not just about the biochemistry of photosynthesis, but you also need to have an understanding of the limiting factors which will change the rate of photosynthesis. So let's take a look at those now. So in general, we can think of the main limiting factors as being these three: light intensity, temperature, and carbon dioxide concentration, although there are other limiting factors which have a lesser effect on photosynthesis. So the rate of photosynthesis, which we can measure in various ways. We can measure, for example, the rate of production of oxygen by a plant, and so on. But the rate of photosynthesis is affected by a number of external abiotic factors. So light intensity, CO2 concentration, temperature, these are all abiotic factors. They are physical factors and chemical factors in the environment which are not involved in living systems themselves until they are incorporated into the process of photosynthesis. So the rate at which photosynthesis happens is limited by whichever of these factors is in the shortest supply at a given time. And this is what we would refer to as the law of limiting factors. Now we can state this as follows: when a process, any process, such as photosynthesis, is affected by a number of factors, then the factor which is in shortest supply at a particular time is going to be the one that determines the overall rate of the process which we're talking about. So you can think of this, or you can think of this in terms of the analogy of a factory production line. Obviously, it's an analogy which falls down in various places, but you can think of it like this. So if you've got a production line which is producing a product such as a car, then the rate at which that production line produces cars is going to be limited by the slowest worker in that production line, or the slowest machine in that production line. And in a similar way, when you've got multiple factors which affect the rate of a particular process, such as photosynthesis, then the rate of that process is going to be limited by whichever of those factors is in the shortest supply. So, for example, at a certain time of day, the light intensity becomes the limiting factor of photosynthesis, because the light intensity will then determine the overall rate of the photosynthesis, because there's only a limited amount of sunlight energy available to drive the light-dependent reactions. Now, if that limiting factor is then increased, the rate of photosynthesis will correspondingly increase. So, the graph here was obtained by investigating the effect of changing the light intensity on the rate of the photosynthesis of a plant. And so we can see here that during this phase, as we increase light intensity to this level, we can see that light is the limiting factor in this region of the graph. But then as we increase the light intensity beyond this point, we get to here where the any further increase in the light intensity does not produce any further increase in the rate of photosynthesis. So an increase in the light intensity, at least initially up to this level, results in an increase in the rate of photosynthesis. But then, at this point, increasing the light intensity to a higher level than this has no effect on the rate of photosynthesis. And so now we can conclude from this that some other limiting factor is limiting the rate of photosynthesis. So increasing the light intensity in general will increase the rate of photosynthesis, but at the very highest light intensities, increasing the light intensity further can have no effect on the rate, because some other factor, such as CO2 concentration or temperature, has become the limiting factor for photosynthesis at the higher light intensities. So let's have a look at what happens when we vary the temperature and the carbon dioxide concentrations on the rate of photosynthesis. Now this is illustrated on the same graph where we are again changing the light intensity and looking at the effect of light intensity on the rate of photosynthesis. So experiment 1, this illustrates that temperature and carbon dioxide concentration become a limiting factor at higher light intensities than this level here. So increasing the temperature from 15 to 25 degrees C in experiment 2 results in an increased rate of photosynthesis at the same light intensities, and also results in a further increase in rate beyond the level of light intensity that was limited in experiment 1. If we then increase the carbon dioxide concentration from experiment 2 to experiment 3, we see a corresponding increase in the rate of photosynthesis in experiment 3. So we see here that both temperature and carbon dioxide concentration have become limiting factors in experiment 1. Increasing either of these factors increases the rate of photosynthesis, with the greatest increase occurring when the value of both of those factors has been increased. So we see here that we increased only the temperature but kept the carbon dioxide concentration the same between experiment 1 and 2, but had we increased both the temperature and the carbon dioxide concentration, we would have produced a much larger increase in the rate of photosynthesis. So we can conclude here that at high light intensities, both temperature and carbon dioxide concentration become limiting factors. Therefore, an increase in either factor results in an increase in the photosynthetic rate. Now, commercial plant growers make use of these ideas to maintain an optimum conditions, a set of optimum conditions for the optimum growth and yield of the plants that they're growing within enclosures such as commercial greenhouses. So we can now use our knowledge of these limiting factors and exploit that knowledge to increase the yield of commercial growth in things like greenhouses. So commercial growers will use artificial lighting to increase the light intensity. Now, these aren't just ordinary lights, these are special lights which are designed to produce those ranges of frequencies which correspond most closely to the absorption and action spectra of the plants which are being grown. Now, it's also important that the light intensity doesn't get too high, because if it does, it can also damage the chlorophyll in the plant leaves. So the amount of light that the plants are given is optimized to the maximum level before that level starts to cause damage. Now also, commercial growers will flood the greenhouses with higher levels of carbon dioxide. Now they can do this by either burning fossil fuels or pumping carbon dioxide gas into those greenhouses. And finally, the commercial growers will raise the temperature of those greenhouses and prevent the night temperatures from falling too low. And this allows the optimum temperature for the growth of the plants. So we see here that the three limiting factors can be, or knowledge of those limiting factors can be exploited by commercial growers in order to produce the optimum rates of growth and commercial yields of fruits, flowers, vegetables, and so on. So let's have a look at another concept that you need to be familiar with, the idea of compensation points. Now, this is something that comes up quite frequently in exam questions, particularly those which involve the interpretation and analysis of data in graphs and in tables. So this is actually quite an important concept for you to master. So, in photosynthesizing organisms, both photosynthesis and respiration involve gas exchange with the environment. Now, during the daylight hours, green plants will do both of those processes, but during the night period, the plants will only respire. So as the light intensity begins to increase from dawn, there comes a point where the rate of photosynthesis is equal to the rate of respiration. Now, the light intensity at which those two processes become of equal rate is called the light compensation point. Now, the time taken for a green plant to reach this point after having been in darkness, this is referred to as the compensation period. Now, at the compensation point, because the two processes of respiration and photosynthesis are happening at an equal rate, it means that the carbon dioxide concentration will remain constant. In other words, there is no net uptake or loss of carbon dioxide by the plant at this point. So, some plants are plants that thrive in conditions where light intensities are very high, whereas shade plants are the opposite. They tend to thrive in dimly lit habitats where they're likely to be overshadowed by taller plants. Now, typical woodlands will show stratification in levels, with the canopy leaves of the tall trees dominating the upper woodland layers, and many of the woodland perennials occupying the lower shaded herb layers of the woodland. So we see here a couple of examples. We see the tall trees, which we can categorize as sun plants. Now, these leaves are photosynthesizing in the upper canopy, the upper high light intensity layers of the woodland, and then way down in the lower layers we find plants like this, oxalis, which is a shade plant which grows and thrives very successfully in low light levels. So, if we investigate the CO2 uptake and output for those two types of plants, we can find data like this. So the rate of photosynthesis, again, equals the rate of respiration where there is no net release or uptake of CO2 by the plant. Now this compensation point occurs at different light intensities for these two plants, but the compensation for the shade plants is much lower than that of the sun plants. So we can see here the two compensation points, again, where there is no net uptake or release of photosynthesis of the CO2 by the two plants. Now, the shade plant, again, begins to photosynthesize at a much lower light intensity than the sun plants, as you might expect. So the compensation period, the time taken to reach the compensation point after having been in darkness for the shade plants, is also much shorter than that of the sun plants. So here we can see the compensation periods. So the ability of shade plants to utilize these low light intensities to power photosynthesis obviously has survival value for these species. Now these species also have various other adaptations to tolerate these dimly lit habitats. So for example, their leaf structure and the fact that they flower and seed early in the year before the dense tree leaf canopy begins to overshadow them. So these are wild garlic plants, and these are adapted to thrive in shady conditions. So these bulbs will flower and seed in the spring once they're planted, before again those overshadowing tree canopies have come into full leaf. Okay guys, well we seem to have come to the end of the first hour of this webinar, so I'm going to give you guys a 5-minute break. After the break we will get into the biochemistry of respiration. So before the break we looked at the biochemistry of photosynthesis. We're going to now look at the biochemistry of respiration. So let me bring that up on the screen here and we will pick up from there. Alrighty, and incidentally everyone, my apologies if there's a little bit of background noise in this particular webinar. It is absolutely boiling hot today in sunny London, and consequently I've had to open the windows because I've not just got the lights on in here, I've got my workstation chugging out heat on my right, and I've got a couple of server cases containing machines in the back also kicking out heat. So it's absolutely boiling, and I apologize if there's a little bit of fan noise or noise from the outside when I've opened the windows here, so hopefully that won't become too distracting for you. Okay, so let's have a look at the biochemistry of respiration here. So we'll begin by looking at glycolysis. So let's have a look at the origin and the significance of glycolysis before we go any further. Now, glycolysis is one of the basic metabolic pathways which is common to all forms of life, both prokaryotic life and eukaryotic life. And this takes place in the cytoplasm, the cytosol of cells, and it has a central role in the process of respiration and in general metabolism. Now, all of that suggests that glycolysis evolved very early in the history of life. Now, the first atmosphere of Earth consisted mainly of carbon dioxide, water vapour, and traces of ammonia and methane and other chemicals. Oxygen was only present in a very tiny, almost negligible concentration, and so the atmosphere of the early Earth was anaerobic in nature. Now, approximately 4 billion years ago, the very first forms of life, prokaryotic life, appeared on the Earth, and these were unicellular chemoautotrophs. They made use of the energy released by chemical reactions in their environments to power their respiratory processes. So these first organisms used carbon dioxide as a source of carbon and oxidized inorganic materials in order to extract that energy. Now later, the prokarya, the bacteria and the archaea, evolved glycolysis, which is a set of chemical reactions which anaerobically free the energy contained within organic molecules such as glucose, and then store that energy in the terminal phosphodiester bond of the ATP molecule. So around 1.5 billion years ago, the prokaryotic cells around at that time, which were related to the archaea, entered into endosymbiotic associations with ancient bacteria to form the ancestral eukaryotes. Now this is part of what we call the endosymbiont hypothesis, and hence glycolysis has evolved as a common metabolic process in both eukarya and prokarya. So it's an anaerobic process. It does not require oxygen, but it can function in both aerobic and anaerobic environments. And so the two major types of the breakdown of glucose, the glucose catabolism, are anaerobic fermentation, in which glucose is partially broken down, and aerobic respiration, in which glucose is completely broken down. So here we see the processes that occur in an aerobic environment, and these are the processes which occur in an anaerobic environment. Now, glycolysis ultimately can occur in either environment. It does not require oxygen, and it is not stopped by the presence of oxygen. So glycolysis, as we can see here, is a precursor process which provides the raw materials which then enter both the aerobic sequence of reactions here and the anaerobic sequence of reactions here. So it can be utilized in both forms of glucose catabolism. Now again, the term catabolism means breaking down, or chemically breaking down, or the processes that involve the chemical breakdown of things. So we start with these processes here. Now we can label the processes that actually produce energy in the form of ATP here. So we've labeled them processes 1, 2 and 3 here. So there are three processes: glycolysis, the citric acid cycle, or Krebs cycle as it's called, and the electron transport chain and chemiosmosis. These three processes directly produce ATP. Now, Krebs cycle, the electron transport chain, and chemiosmosis, these are going to be covered later on in this webinar. But we'll start with glycolysis. Now, in order to fully understand respiration, though, we need to talk a little bit about ATP here. Now, ATP and the structure of ATP is covered in the biological molecules topic and that course module on my website, and also covered in the previous webinar in this series on biological molecules. So ATP forms when chemical reactions release sufficient energy to form a third a bond with a third phosphate group from ADP to an inorganic phosphate. So this is how we form ATP. Now this reaction, this phosphorylation of ADP to form ATP, is reversible. So in other words, we can hydrolyze that terminal phosphodiester bond back to form ADP, an inorganic phosphate, and the release of that energy. So the production of ATP is catalyzed by the enzyme ATP synthase, and ATP is sometimes referred to as the energy currency of cells. It's the main form in which energy is stored and transported to where it's needed within cells. So when there's a demand for energy, the ATP will break down into ADP, an inorganic phosphate, and in so doing will release the energy that was stored in the ATP molecule. Now this process of hydrolysis of ATP to form ADP is catalyzed by an enzyme called ATP hydrolase. So let's look at the formation of ATP. So ATP is synthesized by a condensation reaction in which ADP combines with an inorganic phosphate group, and this in turn produces a molecule of water. And again, this is catalyzed by ATP synthase. So we see here the reaction indicated in this kind of diagrammatic way. So what we're saying here is the energy being used to or the energy being stored in the ATP molecule is actually stored in the phosphodiester bond that forms between the second phosphate group on the ADP molecule and the inorganic phosphate, which forms the third phosphate group in the ATP molecule. So it's really this phosphodiester bond that you see here which stores the energy carried by ATP. Now this is a condensation reaction, so in doing this reaction, in forming that phosphodiester bond, we also form a free water molecule. So this is the high energy bond here. Now the net formation of ATP occurs during respiration and also occurs, as we saw before, during the light dependent stage of photosynthesis. So let's have a look at glycolysis in a little bit more detail. So as we said before, the process of glycolysis occurs within the cytosol of cells. It does not occur within mitochondria. So this usually occurs within the cytosol, and it's a common process which is found in all forms of life. Now, glycolysis, this is, as we said before, a very ancient anaerobic metabolic process which evolved very early in the history of life. And through this process, a molecule of glucose produces 2 molecules of ATP per glucose molecule, but also produces 2 NADH molecules and 2 protons. Now, under anaerobic conditions, the breakdown product of glucose, these 2 pyruvate molecules, can be further converted into either lactic acid in the form in animal cells, for example, or ethanol within plant cells. So, glycolysis is therefore a major pathway for anaerobic respiration because, again, this does not require an input of oxygen. So, under aerobic conditions, the pyruvate undergoes further chemical catabolism via the Krebs cycle, or the citric acid cycle as it's sometimes called, and this yields 2 ATP directly per original glucose molecule, as we'll see. And also later on we'll see how the electron transport chain and the process of oxidative phosphorylation will generate an additional 34 ATP per original glucose molecule. So this gives a net yield of 38 ATP per glucose molecule under aerobic conditions. Again, we'll see the details of that shortly. So let's start by making reference to the structure of a glucose molecule. Now again, in the previous webinar on the subject of biological molecules, we go into the structures of monosaccharides and also carbohydrates, complex carbohydrates in more detail, but for the moment it's enough to make reference to the structural formula that you see here. Now we can simplify this for the purposes of this discussion and looking at what happens to the glucose molecules in a very similar way to the way that we simplified the structures of the molecules involved in the processes of photosynthesis. So rather than use this structure, we will simply represent a glucose molecule as a 6-carbon chain, very much like this. So the first step of respiration is that glucose is said to be primed with energy. So this involves essentially the transfer of chemical energy from an ATP molecule to a 6-carbon glucose molecule. Now, in doing this, what we're essentially doing is we are phosphorylating the glucose molecule at carbon 6, this position here on the glucose molecule. Now, in doing this, we are expending energy, but we are also forming a bond to an inorganic phosphate group. So by doing this, we have gone from a glucose molecule to a glucose 6-phosphate. Now again, the 6 in this name refers to the position of the phosphate group on the carbon chain which makes up this molecule. So by convention, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5 and carbon 6. Carbon 6 is the carbon which is kind of outside of the 6-membered ring in the glucose molecule. So we've gone from glucose to glucose 6-phosphate by the expenditure of a single ATP molecule to provide energy for the process, to prime the glucose molecule with energy, and also the use of the phosphate which was hydrolyzed off the ATP molecule and the bonding of that phosphate to carbon 6. So we've formed glucose 6-phosphate here. So the glucose molecule requires further activation before it starts to split apart. Now, this occurs again at the expense of an ATP molecule. So the glucose 6-phosphate first has to be rearranged. It has to be isomerized to form a fructose 6-phosphate molecule. So we've gone from a 6-membered ring sugar to a 5-membered ring sugar with an additional carbon here and an additional carbon here. So what we've done here is we've still got a 6-carbon sugar, but it has a 5-membered ring here rather than a 6-membered ring. So essentially what we've done here is we have reorganized the structure of the glucose 6-phosphate molecule to form a fructose 6-phosphate molecule, and in order to do that we need again another ATP molecule. So this is called an isomerization because we haven't removed or added any atoms to this molecule to form this molecule. We have simply reorganized the bonding between the existing atoms in the molecule. So we now have a fructose 6 molecule where we have a phosphate group located on carbon 6, but we also have carbon 1 now located outside of the ring. So now a phosphate group can now be transferred from ATP to carbon 1 of the fructose 6-phosphate, and this further activates the molecule. It raises its potential chemical potential energy. So we've gone now, by the addition of a further ATP molecule, we have now phosphorylated carbon 1 as well. So in doing this, we have now formed fructose 6-bisphosphate, or fructose 1,6-diphosphate. Now this molecule is sufficiently activated, in other words it has a sufficiently high chemical potential energy that it becomes unstable, and it stabilizes itself by splitting into two separate, more stable molecules. So the conversion of glucose to fructose 1,6-bisphosphate or diphosphate, which sometimes you can refer to more simply as a hexose phosphate, this activates the glucose, and again the process whereby we go from glucose to fructose 1,6-bisphosphate requires in total the use of 2 ATP molecules. So we've gone from glucose to glucose 6-phosphate by the use of 1 ATP molecule, and we also go from glucose 6-phosphate by an isomerization to fructose 6-phosphate. And then we go on to further phosphorylate the fructose 6-phosphate using another ATP molecule to fructose 1,6-phosphate. So this activates the molecule. Again, we are going from successively lower energy stable molecules to more and more unstable high energy activated molecules. And now this molecule is sufficiently activated to break down to form your two molecules, which we'll see, two more stable molecules, which we'll see shortly. Now, it's important here that we need to mention that the direct phosphorylation of any substrate molecule is called substrate level phosphorylation. This is in contrast to, as we'll see, other types of phosphorylation, which we can refer to as well. So this phrase, substrate phosphorylation or substrate level phosphorylation, refers to the direct phosphorylation of a substrate molecule using the hydrolyzed ATP molecule and the inorganic phosphate from that hydrolysis. So now we have our activated high potential energy fructose 1,6-diphosphate molecule, which then breaks apart. Now we call this cleavage. So this molecule cleaves into two separate molecules of 3 carbon phosphates. So these are called triose phosphate molecules. We form 2 triose phosphates from the original fructose 1,6-bisphosphate, because again this is a hexose sugar, it has 6 carbons, but when it breaks apart it forms 2 3-carbon sugars, both of which are phosphorylated. Now to visualize this, again we can illustrate like this. So we started with a 6-carbon sugar, we ended up with a diphosphorylated 6-carbon sugar, fructose 1,6-bisphosphate. Again, that took 2 ATP molecules, but now that we have this activated molecule, it's unstable and it breaks apart to form 2 3-carbon phosphorylated sugars, triose phosphates. So each triose phosphate molecule is now converted into pyruvic acid or pyruvate by a series of steps. So first, each of these molecules is converted into a phosphoglycerate molecule or GP, and this is an oxidation which involves the removal of hydrogen atoms from each of these 2 triose phosphate molecules. So by doing this, we liberate 2 hydrogen atoms per molecule. Now molecules of ATP are actually formed during this reaction, so this is a direct formation of ATP by using the energy released when this triose phosphate molecule forms the phosphoglycerate molecule. So not only do we have 2 molecules of ATP being formed, we also have 4 hydrogen atoms being released by these 2 processes. So glycerate phosphate is now converted into pyruvate or pyruvic acid, and this actually involves, again, the release of sufficient energy to form another molecule of ATP per phosphoglycerate molecule, which is converted into pyruvic acid. Again, there are 2 of these happening per single glucose molecule that we started with. Now the 2 hydrogen atoms which are released by each of these conversions are accepted by a molecule of a hydrogen carrier that we saw before, NAD+, to form NADH2. Now due to the prevailing pH, the NADH2 dissociates to form NADH and a single proton. So let's summarize glycolysis then. So one molecule of glucose is primed with energy and forms fructose 1,6-bisphosphate, and this utilizes a total of 2 ATP molecules. This fructose 1,6-bisphosphate is then broken apart or cleaved into 2 molecules of triose phosphate. Each triose phosphate is then oxidized to form a molecule of pyruvic acid, and this stage generates a total of 4 molecules of ATP. Now the net gain of ATP from glycolysis is therefore just 2 molecules of ATP, because we used up 2 molecules but we generated 4, so the net gain is 2 molecules of ATP per molecule of glucose that forms through this that is used in this process. Now in addition to that, 2 molecules of reduced hydrogen carriers, 2 NADH, and also 2 hydrogen ions are produced in total in this process. So, before we go on to the anaerobic phase of respiration, let's have a look at some different forms of anaerobic respiration. So again, let's take a close look at the role of ATP. Now again, the energy produced by anaerobic respiration is primarily stored and released by that energy currency molecule ATP, just the same way it is in aerobic phases of respiration. So ATP forms again when the chemical reactions involved release sufficient chemical energy to bond that third phosphate group to the ADP molecule. So this is reversible. The forward reaction to produce ATP is catalyzed by ATP synthase, and the backward reaction is catalyzed by ATP hydrolase. So this is what we've seen before, and again, ATP is the energy currency of all living systems. So again, we saw that glycolysis is a cytosolic process. It occurs within the cytosol. Now, under anaerobic conditions, we can either form lactate, which is seen in animal cells, but we can also see the formation of alcohols such as ethanol in things like plant cells. So let's have a look at these anaerobic pathways in a bit more detail now. So some living organisms will derive their energy by breaking down sugars in the absence of oxygen. So these are what we would refer to as anaerobes, and the biochemical processes involved in this form what we call anaerobic respiration. Now among the anaerobes, some organisms are classed as obligate or complete anaerobes because they can only thrive in oxygen deficient conditions. So an example of this would be the Clostridium tetani bacteria. This is an obligate anaerobe, and it's a common soil bacterium. Now this multiplies under the conditions in the soil which are very low in oxygen. Now these are the organisms responsible for the disease tetanus, and when this organism enters wounds, this can then enter the bloodstream and then cause that disease. And it does so by the release of a toxin. Now, the toxin actually interrupts the transmission of impulses to the skeleton, and this leads to sustained muscular contraction. And this is referred to as tetany of the muscles. So the sustained muscular contraction in the disease tetanus, this is illustrated by a drawing made of an afflicted soldier during the Napoleonic Wars, and typically this is what you see in advanced cases of tetanus. The axial musculature of the back and the spine is so extremely contracted that their back will bend to this extreme angle. And if it occurs in infants, the infants typically will also show this muscular contraction of all their muscles. So yeast is a single-celled fungus. Now this is able to resort to anaerobic respiration when the oxygen is in absent or short supply. So yeast does thrive, however, in aerobic conditions as well. So unlike the previous organism, the Clostridium tetani, this is classed as a facultative or partial anaerobe. So in other words, it doesn't have to exist only in anaerobic conditions. It can respire anaerobically when it requires, when it finds itself in anaerobic conditions, but it can equally thrive under aerobic conditions. So yeast cells can respire both aerobically and anaerobically, and as such these organisms are referred to as facultative anaerobes. Now plants and fungi can carry out another form of anaerobic respiration, and the products produced by that are ethanol and carbon dioxide gas. Now this is an incredibly important commercial process because it's the basis of all baking, brewing and distilling industries. So the overall equation for this form of anaerobic respiration as a word equation would be this, and as a chemical equation it would be this. Now generally speaking, I don't like to put energy into chemical equations because strictly speaking energy is not a substance, so it shouldn't appear as a reactant or a product in a chemical equation, but I'm simply kind of putting it here just to remind you that these processes are exergonic, they release useful energy. So the glucose will initially undergo glycolysis to produce pyruvate, but then under anaerobic conditions the pyruvate is not converted into the broken down all the way to carbon dioxide as it would be in the Krebs cycle, because there isn't enough oxygen available. Instead it is broken down to form alcohols. So this process here is illustrated here. So under anaerobic conditions, in other words, under conditions of alcoholic fermentation, organisms like yeast will take the pyruvate formed by glycolysis and then convert it first into an aldehyde called ethanal, and then ultimately convert that aldehyde into the alcohol ethanol. But this anaerobic pathway does regenerate the NAD+, which can then drive further glycolysis like this. So in plant cells and fungi, the end product of anaerobic respiration is an alcohol, ethanol, and in these cases one molecule of glucose is converted by glycolysis again into two molecules of pyruvic acid, yielding 2 ATP, 2 NADH and 2 H+. Now the pyruvic acid is then converted into this aldehyde, ethanol, and 2 molecules of it, because we start with 2 molecules of pyruvic acid, and also this produces CO2 as a byproduct of the process. Now the reason for this is because we're starting with a 3-carbon compound and we're ending with a 2-carbon compound, and this is where the CO2 comes from. Now each ethanol molecule is then reduced further by NADH and protons to form ethanol, and in doing this, this yields NAD+. Now the NAD+ produced by this is then cycled back and used again in glycolysis. So during this entire process of alcoholic fermentation, there is a net gain of 2 ATP molecules per glucose molecule. So this process is fundamentally important, as we said before, in the production of alcohol for breweries and distilleries, but it's also important in the baking industry. So remember that ethanol here is referred to as acetaldehyde. This is the chemical name for ethanol, but this is also a valid chemical name as well. So we said that the alcoholic fermentation of yeast is also important in bread making. Now the reason for this is not because of the production of ethanol, but the production of carbon dioxide by the yeast, and this is what produces the bubbles of gas inside the bread, which causes the dough to rise. But also obviously it's important in the production of alcoholic drinks. Now we can also produce vinegar as a product of this process. So this is made by bacterial fermentations of solutions that already contain ethanol, which has already been produced by alcoholic fermentation in yeast. Now acetic acid bacteria will oxidize the ethanol to form acetic acid or ethanoic acid, and ethanoic acid is the active ingredient which gives vinegar its flavour. Now the acetic acid in table vinegar is somewhere between 4 to 8% by volume and up to 18% for pickling vinegars. So vinegar can be made from many sources. We can make it from wine, beer, cider, rice, pretty much anything that has ethanol in it, and this is used for flavourings and also for preservatives. So we mentioned alcoholic fermentation to produce alcohols, but anaerobic fermentation, or anaerobic respiration rather, occurs within most living cells and involves the breakdown of food molecules in the absence of sufficient oxygen. Now, unlike fungi and plants, in animals, humans and other animals, this can occur within muscle cells, and the immediate product of this is lactic acid. Now in prokaryotes, these can also respire anaerobically, and in this group the immediate product can also be lactic acid. Now the overall equation for anaerobic respiration in bacteria and in animals is this. So as a word equation we can write it like this. As a chemical equation we can simply write it like this. So the glucose again will initially undergo glycolysis to produce pyruvate, but then the pyruvate does not go into the Krebs cycle because the Krebs cycle requires an input of oxygen. Instead, it is converted into lactic acid. So certain species of bacteria which are found naturally in milk will convert the milk sugars or lactose into lactic acid. Now if a mixture of milk and lactic acid bacteria are kept at an optimum temperature of around 45 degrees centigrade, the enzyme lactic acid produced by the bacteria can act upon the lactose to produce lactic acid. Now it's this lactic acid which will denature the milk protein casein, and this causes the milk to coagulate. The milk then thickens and curdles, and this then turns that milk into yogurt. Now another important product of these types of processes is the production of cheeses. So the anaerobic production of lactic acid by certain types of bacteria, this is actually what gives many cheeses their flavour. Now the large variety of the tastes and textures is due to the degree to which the curd has been dried and compressed, and also the origin of the milk and the variety of the bacteria and fungi which have been used in the maturing process. So the further processing of pyruvate in lactic acid fermentation, this does not actually produce any more ATP than glycolysis alone. However, this anaerobic pathway does regenerate NAD+, which can then drive further glycolysis. Now the enzyme lactate dehydrogenase will then catalyze that production. So in animal cells, such as human muscle cells and certain species of bacteria, the end product, as we said before, is lactate or lactic acid. And in these cases, one molecule of glucose is converted into 2 molecules of pyruvic acid during glycolysis, and this also yields 2 ATP and 2 NADH and 2 protons directly, but then each pyruvic acid molecule is then further reduced by NADH and H+, apologies for that siren in the background, and in so doing it yields NAD+. Now, those 2 molecules of pyruvic acid are then converted into lactic acid, and then the NAD is then used again to drive further glycolysis. Now, during this process, there is a net gain of 2 ATP molecules per glucose molecule. So, aerobic respiration in muscle cells, we can summarize at least the first stage of this, anaerobic respiration rather, in muscle cells with this diagram as we've been discussing these processes here. Now it's the buildup of lactic acid within the muscle cells which ultimately leads to muscle cramps and muscle fatigue. So yeast cells can carry out respiration in 2 ways, either aerobic or anaerobic, because as we said before, they are facultative anaerobes. If oxygen is present, then they respire aerobically. If oxygen is absent or in low concentration, it respires anaerobically, and in so doing produces carbon dioxide and alcohol. Now anaerobic respiration provides less energy per unit of glucose than aerobic respiration, and again aerobic respiration is so much more effective at producing ATP that in fact the yeast requires aerobic conditions in order to grow and reproduce, even though it can operate under anaerobic conditions. So, anaerobic respiration in cells such as yeast cells, muscle cells, certain bacteria, again, this yields only 2 molecules of ATP per glucose molecule respired. Now, the ATP yield is produced exclusively by the glycolytic stages of that process. In other words, once you reach the end of glycolysis, if you're under anaerobic conditions, you've produced 2 ATP molecules per glucose molecule, but if you remain under anaerobic conditions, that's all you're going to get. So, the complete aerobic breakdown of glucose through the full process of aerobic respiration produces 38 molecules of ATP per glucose molecule respired. So this gives you a sense of just how important and how advantageous aerobic conditions are to a facultative anaerobe. So there's a clear advantage for facultative anaerobes to switch to aerobic respiration and away from anaerobic respiration whenever conditions will permit them to do so. So again, the 3 processes that directly produce ATP are labelled here. And each molecule of glucose in an anaerobic environment would only produce 2 ATP molecules. So in other words, stage 1 only, because you require aerobic respiration for the direct production of ATP at any other stages here. But however, if you do have oxygen available in sufficient quantity, the organism can continue to respire and would produce 38 ATP molecules using stages 1, 2 and 3, involving both direct production of ATP and also production of ATP by the electron transport chain and chemiosmosis. Now in contrast, we see here that the anaerobic pathway only produces 2 ATP molecules per molecule of glucose. So, facultative anaerobes can switch from aerobic to anaerobic depending on what the requirements are or what the environmental conditions are. Now, typical conditions which will result in that switch are insufficient oxygen to maintain aerobic respiration, or if the pyruvate cannot be processed quickly enough or at all. So an example of this is the ability of higher plants to switch to anaerobic respiration when their roots become waterlogged or submerged. Now another example of this is in humans when you undergo vigorous exercise. So for example, sports such as the 100-meter sprint, almost the entire time the athletes are actually respiring anaerobically because their bodies are generating pyruvate at a rate which is too high to be broken down by the available oxygen, given even their highly trained respiratory system's ability to deliver that oxygen to their respiring tissues, along with their circulatory system. So each physical activity or sport that's undertaken require different energy systems. Some will mainly use aerobic respiration, some will use mainly anaerobic respiration, but most sports will use some combination of the two. So basketball players will use both, for example. But in terms of track events, their use of anaerobic respiration, we can kind of summarize using this table here. So marathons use almost zero, marathon runners will use almost 0% anaerobic respiration. The 10,000 meter run uses about 5%, 5,000 meter runners use about 7%, 1,500 meter runners about 40%, 800 meter runners about 50%, and then you see the pattern that emerges here until you get to the 100-meter sprint, where it's more than 99% anaerobic respiration. So this links to an idea that we call the oxygen debt. So during vigorous exercise, the human skeletal muscle cells can respire anaerobically, but only for short periods of time. Now the switch to anaerobic respiration and the subsequent buildup of lactic acid is usually due to a shortage of oxygen at the cellular level. Now the lactic acid is what is responsible for your muscles beginning to feel tired and painful. Now some people refer to this as the stitch which you feel in your sides if you're running. Now this is actually a muscle cramp which occurs as a result of the muscle not receiving sufficient oxygen to fully break down the pyruvate produced by glycolysis. So lactic acid is toxic, and its future breakdown will require an oxygen-rich environment at the cellular level. Now this future need for oxygen to break down the lactic acid that built up during that heavy exercise, this is referred to as the oxygen debt, and this is the oxygen debt that will be repaid once the exercise has been finished. So this is why athletes who undergo vigorous exercise like the 100-meter sprint, at the end of the sprint you'll see them bending over with their hands on their knees, breathing very heavily. What they're doing is they are repaying the oxygen debt by absorbing large quantities of oxygen into their bloodstream, delivering that to their muscle tissues to break down that pyruvic acid and that lactate that is built up in those tissues, all the way down to carbon dioxide and water, and releasing ATP. So this is what you will see. Now, the removal of lactic acid from your tissues, this is achieved by the action of the enzyme lactate dehydrogenase. Now this catalyzes the conversion of lactate back into pyruvate, and then the pyruvate then is incorporated into the Krebs cycle. Now 70% of the lactic acid is oxidized into pyruvate by lactate dehydrogenase, 20% of it is converted into glucose, which can then be used for energy in the liver, and then the final 10% is converted into protein. So for facultative anaerobes, during aerobic respiration, the operation of the electron transport chain, as we'll see, is dependent upon the ability or the availability of oxygen to function as the final electron acceptor, which again, the electrons are passed down the electron transport chain to successively lower energy levels, but final acceptor of those electrons is the oxygen molecule, and in doing so it combines with two protons to form the water molecule, and this is in fact the origin of the production of water in respiration. Now when oxygen is unavailable, there is no final acceptor for the electrons being passed down the electron transport chain by the reduced carrier NADH. So again, if there is no oxygen to accept that final electron, the electrons will remain attached to the last carrier in the chain, which is now in a reduced state. So as more electrons pass along the chain from further molecules of NADH, they are unable to pass to the last carrier, which is already in a reduced state. Now the consequence of this is that the electrons then remain attached to these carriers, and again those carriers remain in a reduced state. So this essentially creates a domino effect, which effectively stops any further hydrogen atoms being accepted to form NADH. So we now have basically all of the electrons in the electron transport chain ultimately becoming in the reduced state, and this in turn basically holds up the entire process. Now remember, the majority of the ATP which is formed per glucose molecule under conditions of aerobic respiration are formed as a result of this electron transport chain and chemiosmosis. So if you're not having this happening because of this kind of domino effect, then ultimately you are not producing the majority of the ATP molecules that you could produce under aerobic conditions. So all of the electron transport chain carriers are now in a reduced state, so no further pairs of hydrogen atoms can be accepted by the chain. The NADH molecules are unable to be oxidized back to form NAD+, and so as molecules of NAD+ within the mitochondria now become unavailable, then the Krebs cycle then ceases to operate. So let's have a look at the aerobic phases of respiration, which involve both the Krebs cycle or citric acid cycle and the electron transport chain. So aerobic respiration occurs within most living cells, and it's the process by which food molecules, principally glucose, are broken down in the presence of oxygen to generate energy, useful energy for the cell. Now again, the overall equation for aerobic respiration that you're familiar with is this, but as we saw, this is actually a gross simplification of a multi-step process that occurs within cells. So molecules of ATP store much of the released energy, and some of the energy released is lost as wasted heat. Now this is actually the source of body heat in mammals, for example. So we mentioned the role of ATP, so I'm going to skip past this. And actually I see that we have we've come towards the end of the second hour of this webinar, so before I get into the Krebs cycle and the aerobic phases of the Krebs cycle, the electron transport chain and chemiosmosis, I'm going to give you guys another 5-minute break. So to remind you, under ordinary conditions, normal aerobic cellular respiration is the utilization of oxygen by cells for the production of ATP. So the aerobic phases of respiration is made up of a series of over 20 chemical reactions, and these can be divided up into 4 general phases. So the first phase is glycolysis. Now again, glycolysis itself is anaerobic in nature. It occurs in the cytoplasm of all living organisms. So this occurs in the cytoplasm and does not require oxygen, and it involves the breakdown of one molecule of glucose, which is a hexose sugar, a 6-carbon sugar, ultimately to form 2 molecules of 3-carbon sugars, pyruvic acid or pyruvate, and during this process of glycolysis there is a net production of 2 ATP molecules. So this occurs in the cytoplasm or the cytosol. Now glycolysis is also a major part of the anaerobic or plays a major role in the anaerobic respiration, because under anaerobic conditions pyruvate can be converted into either ethanol in plants and fungi or lactate in animals. So we mentioned this in alcoholic and lactic acid fermentation. So in prokarya, such as bacteria, so again, these lack mitochondria. So all the phases of respiration are performed in the cytoplasm. Now the proton gradient for ATP production is across the cell membrane of a bacterium, because they do not have internal membrane-bound organelles. Now in contrast to that, in eukaryotic cells, as we'll see, the aerobic phases of respiration, including the electron transport chain and chemiosmosis, this occurs across the cell membrane of the mitochondria. So phase 2 is called the link reaction, and in eukaryotic cells it takes place within the fluid matrix of the mitochondrion. So pyruvic acid, which is the product of glycolysis, then diffuses into the fluid matrix of the mitochondrion, where it's converted into a 2-carbon compound called acetyl coenzyme A, or acetyl CoA for short. So this phase generates some carbon dioxide. So here we see what's happening here. So the anaerobic phase of glycolysis produces 2 pyruvate per glucose molecule. Those pyruvate molecules then diffuse across the 2 membranes of the mitochondrion, the outer membrane and the inner membrane, into the fluid matrix in the center of the mitochondrion. There, the pyruvic acid molecules are converted into 2 acetyl CoA molecules, and in so doing, this releases carbon dioxide. So the 2-carbon acetyl CoA compound is then needed to drive the next phase of the aerobic side of respiration. So phase 3, this is the first truly aerobic phase because it requires oxygen. This is called the Krebs cycle, or the citric acid cycle, and this takes place again within the fluid matrix of the mitochondrion. Now it is a cycle of biochemical reactions, and we're going to talk about that in more detail shortly, but essentially this involves the stepwise oxidation of the 6-carbon compound citric acid ultimately into a 4-carbon acid. So in this case, the oxidation involves removal of pairs of hydrogen atoms. Again, remember the definition of oxidation and reduction with respect to hydrogen atoms. Oxidation is loss, reduction is gain. So we remove pairs of hydrogen atoms from these acids in the cycle, and in so doing, carbon dioxide gas is also generated. So 2 molecules of ATP are also formed from 2 turns of this cycle directly. So phase 4 is called the electron transport chain, and this takes place on the cristae, these infoldings of the inner mitochondrial membrane. So in this phase we have pairs of hydrogen atoms which were generated during the earlier phases of respiration, and these are carried to these cristae. So the electrons from these hydrogen atoms are then passed along a chain of carrier molecules, and these carrier molecules themselves are part of the inner mitochondrial membrane along these cristae. Now again, this is a stepwise release of energy as these electrons are transported, and each time the electrons are transported from one carrier to the next, because the first carrier is at a higher energy level to the second carrier, the difference in energy is used to directly drive the formation of ATP from ADP. So phase 4 is called the electron transport chain. Now in this phase, oxygen, as we said before, needs to be present because oxygen is the terminal electron acceptor at the end of the chain, and when it accepts electrons, it also combines with 2 hydrogen ions to form a water molecule. So this mechanism for producing ATP is called oxidative phosphorylation, in contrast to substrate level phosphorylation, which is the direct production of ATP from the chemical energy released from the chemical reactions of producing or converting one substrate into the next. In contrast, we have oxidative phosphorylation, which is the production of ATP which is driven by the energy released from successive oxidations of the carrier molecules in the electron transport chain. Now, as we said before, this is actually the main source of the majority of the ATP produced under aerobic conditions from glucose. Now, 34 of the 38 ATP per molecule of glucose produced in aerobic respiration is actually produced by this oxidative phosphorylation process. So oxygen is required because oxygen is the oxygen atom is the final electron acceptor. It accepts 2 electrons and also at the same time bonds with 2 protons, ultimately to form the water molecule. So, as an overview, we can say that 1 molecule of glucose is oxidized to 2 molecules of pyruvic acid during glycolysis. This occurs in the cytosol and occurs both under aerobic and anaerobic conditions. Now, under aerobic conditions, the main product is pyruvate. Under anaerobic conditions, the main product is lactate. Now, in aerobic respiration, the pyruvate molecules then diffuse into the fluid matrix of the mitochondria. In animals, the pyruvate is actively taken up by the mitochondria. The 2 pyruvate molecules are then converted into 2 2-carbon molecules of acetyl coenzyme A in what we call the link reaction. The acetyl coenzyme A molecules are then fed into the Krebs cycle, and Krebs cycle involves successive oxidations of acids, organic acids, and this generates pairs of hydrogen atoms, also carbon dioxide, and also some ATP by substrate level phosphorylation in this process. Now, the pairs of the hydrogen atoms are then carried to the cristae of the mitochondria, the inner infoldings of the inner mitochondrial membrane, and there they pass electrons from the hydrogen atoms along the carriers in that chain. Now, the final electron acceptor is molecular oxygen, and this combines along with those electrons with protons to form water molecules. So 38 molecules are formed per molecule of ATP under aerobic conditions by the whole series of processes, all the way from glycolysis through the link reaction to the Krebs cycle through to oxidative phosphorylation and chemiosmosis. So that's an overview. Let's take a close look at the link reaction. So pyruvate molecules are formed by glycolysis. They diffuse into the fluid matrix of the mitochondria. Now again, in animal cells, the pyruvate molecules are actively transported into the mitochondria because they're being transported against their concentration gradient. So each 3-carbon pyruvate molecule is converted into 2-carbon acetyl-CoA. Again, to visualize this, we can use this diagrammatic representation here. Now, the pyruvic acid is oxidized to acetyl-CoA through the removal of a pair of hydrogen atoms. So we now use those hydrogen atoms to reduce NAD to form NADH plus, ultimately NADH plus H plus. So in doing this, in converting the 3-carbon compound to a 2-carbon compound, we also release a carbon dioxide molecule. So this is called decarboxylation. Now, the 2-carbon acetyl group is then needed to drive the process of Krebs cycle. So coenzyme A is recycled, as we can see here, and the coenzyme A is recycled back into this process of the link reaction to once again form more acetyl-CoA. So the link reaction overall yields a 2-carbon acetyl group, and again this is needed to drive the Krebs cycle, and also yields the reduced hydrogen carrier, NADH, also a proton and also a carbon dioxide molecule. So let's look at Krebs cycle now. So we start with acetyl-CoA, the 2-carbon compound. Now this combines with a 4-carbon acid called oxaloacetic acid, and this forms a 6-carbon compound, citric acid. So in this process, the coenzyme A is recycled, and the 6-carbon citric acid is then oxidized to a 5-carbon compound called alpha ketoglutaric acid. In doing this, this also involves the removal of hydrogen atoms from the 6-carbon acid, and these are then accepted by the hydrogen carrier NAD+ to form NADH and a proton. Now the number of carbon atoms has been decreased here from 6 carbons to 5 carbons, and of course this will involve decarboxylation to form a molecule of CO2. Now at this point, the 5-carbon acid is then oxidized to a 4-carbon acid called succinic acid. And this in turn generates another NADH2 molecule, which then further breaks down to form NADH plus H+. We also are removing a carbon by decarboxylation, so we are generating another carbon dioxide molecule. And we are also forming directly a molecule of ATP by substrate level phosphorylation. Now the 4-carbon succinic acid is then further oxidized to a 4-carbon malic acid. Now this step, the removal of hydrogen involves the addition of hydrogen to a different hydrogen carrier, FAD, flavine adenine dinucleotide, and this forms FADH2. So in the oxidation of succinic acid to malic acid, we are removing a pair of hydrogen atoms. That pair of hydrogen atoms is accepted by this hydrogen carrier, FAD, to form FADH2. Now the final step is that the 4-carbon malic acid is oxidized back to reform 4-carbon oxaloacetic acid, and this enables the whole cycle to occur again, but in doing this we are generating more protons, which can be, or rather more hydrogen atoms, which can be further accepted by NAD+, ultimately to form NADH plus additional proton. So this is a kind of overview of the Krebs cycle, or the citric acid cycle. So one turn of Krebs cycle directly generates 3 molecules of NADH and 3 protons, 1 molecule of FADH2, 1 molecule of ATP directly by substrate level phosphorylation, 2 molecules of carbon dioxide. So because 1 molecule of glucose generates 2 molecules of acetyl-CoA, this means that for every 1 molecule of glucose that goes into aerobic respiration, you get 2 turns of this Krebs cycle. So you get 2 lots of all of these products. So let's summarize Krebs cycle then. So this involves the stepwise oxidation of 6-carbon citric acid into ultimately the 4-carbon oxaloacetic acid, and this involves the removal of pairs of hydrogen atoms from each of these acids that are being oxidized. Ultimately, it's the hydrogen carriers NAD+ and FAD which accept pairs of hydrogen atoms, and in turn they are reduced as a result. Now ATP is generated during the cycle directly by substrate level phosphorylation. Carbon dioxide is also produced as a waste product, which is formed when we decrease the number of carbon atoms in each of these steps. So one turn of the cycle, as we said before, generates 3 molecules of NADH, 3 protons, 1 molecule of FADH2, 1 molecule of ATP by direct substrate level phosphorylation, and 2 molecules of carbon dioxide. However, for every 1 glucose molecule, remember, we generate ultimately 2 molecules of acetyl-CoA, and so we get 2 turns of the citric acid cycle. So in addition to pyruvate, and again this is the breakdown product, the ultimate breakdown product of carbohydrates, there are other respiratory substrates so there are other respiratory substrates which can be used to power the Krebs cycle. So this is why proteins and fats and oils have a high energy value. It's because ultimately in the absence of carbohydrates, these other substrates, proteins and fats, can be broken down, and then the products of that breakdown fed into the citric acid cycle or Krebs cycle in order to generate ATP. So let's finish off here by just mentioning a little bit more about that. Now again, this is not necessarily something that you need to know in detail. The actual biochemical pathways by which these conversions of amino acids and glycerol and fatty acids from lipids are converted into these intermediates in the link reaction and acetyl-CoA, these are rather complicated series of biochemical reactions. You don't necessarily have to know the full routes, but this is a kind of summary at the level that you probably need to know for advanced level currently. So what we're saying here is, in the absence of glucose, or in the absence of carbohydrates that can be broken down to glucose, there are other sources of the intermediates involved in these biochemical pathways, which can be used to power the production of ATP. Now, specific amino acids will enter via just one of the 4 numbered routes, route 1, route 2, route 3, into the respiratory system. So, for example, alanine will enter via route 1. Alanine will be converted into pyruvate. Leucine will be enter route 2. Leucine can be converted into acetyl-CoA. Aspartic acid will enter via route 3 and will be converted into oxaloacetate, which can then feed the Krebs cycle, and so on. Glutamic acid, route 4, etc. So let's have a look at the electron transport chain now. Now, we said that the electron transport chain involves carrier molecules which accept and then pass on electrons to other carrier molecules, and these carrier molecules are located in the inner mitochondrial membrane in these folds which we call cristae. So we see here an electron micrograph of a section through a mitochondrion where we can clearly see these infoldings of the inner mitochondrial membrane, and this is where the chains of carriers are located. Now, the carriers are conjugated proteins called cytochromes, and many of them are cytochromes, and these are arranged within the cristae membranes, and again they are arranged in order of decreasing redox potentials or decreasing energy levels for the electrons. Now, ultimately it's the reduced carriers NADH and FADH2 together with protons which are formed during glycolysis, the link reaction and Krebs cycle. Now, these effectively transport pairs of hydrogen atoms to this electron transport chain, and it's the electrons carried by those hydrogen atoms which then pass down this chain, and the energy differences between each of those successive oxidations and reductions, these are then used to drive the production of ATP by oxidative phosphorylation. So NADH is the first carrier, and it transfers hydrogen atoms to the second carrier in the chain. So NADH in doing this has become oxidized back to NAD+, and the second carrier in turn has become reduced. So this is a redox reaction because it's a reaction in which one species has been reduced and another species has been correspondingly oxidized. So a pair of hydrogen atoms is then passed to the third carrier in the chain, which in turn becomes reduced, and in doing this the second carrier is said to be oxidized back to its original state. So this is another redox reaction. So as the hydrogens flow from the second to the third carrier, sufficient energy is released to build a molecule of ATP from ADP and a free inorganic phosphate. So here we have produced an ATP molecule. Now, each hydrogen atom then splits into its respective electron and hydrogen ion and proton. So this is indicated by this equation here. So now the hydrogen is split into two hydrogen atoms split into 2 protons and 2 electrons. Now the electrons then continue to be passed down the chain of carriers like this, whereas the molecular oxygen becomes the final electron acceptor. Now the hydrogen ions, the protons, will then unite with the electrons and the molecular oxygen to form water molecules at this energy level. So as the electrons flow down this chain from carrier to carrier, sufficient energy is released between these or by these redox reactions to build a further 2 molecules of ATP. So again, we are talking about what happens ultimately when one molecule of NADH plus H plus passes its 2 hydrogen atoms to this first carrier in the chain. So sufficient energy to build ATP molecules is generated therefore at 3 sites in this electron transport chain on the way down all the way to the final acceptor, molecular oxygen. So for each NADH molecule plus H plus produced during glycolysis and the link reaction and Krebs cycle, you get 3 molecules of ATP formed by this process of oxidative phosphorylation through the electron transport chain. So the carriers forming the transport chain are first reduced, then reoxidized as the hydrogen atoms and then the electrons flow down the chain. So a series of redox reactions are occurring along the length of this chain, because each time one of these carriers releases an electron, it has become oxidized, whereas the next carrier correspondingly has become reduced because it has gained electrons. So FADH2 is the other hydrogen carrier that delivers pairs of hydrogen atoms to the electron transport chain. Now, due to its redox potential, FADH2 delivers pairs of hydrogen atoms to a carrier further down the chain than NADH in terms of its energy level. So when we're talking about the electron transport chain, we fundamentally have a couple of different hydrogen carriers which are delivering protons or delivering hydrogen atoms to this chain. So when FADH2 delivers its pair of hydrogen atoms to the electron transport chain, sufficient energy is generated this time to form two molecules of ATP. So what we're saying here is that NADH, for every NADH plus proton, which feeds its two hydrogen atoms at the top level of the chain, at this energy level up here, ultimately as the electrons are passed down the chain, you get three molecules of ATP formed by oxidative phosphorylation per molecule of NADH plus H+. But for FADH2, it will insert its electrons and protons at a lower energy level. So for every molecule of FADH2 that you get from, for example, a Krebs cycle, you will only produce two ATP molecules as those atoms and electrons pass down that chain to the final electron acceptor. So the thing to remember here is that NADH generates 3 molecules of ATP by oxidative phosphorylation, whereas FADH2 generates 2 molecules of ATP per molecule. So the reduced carrier molecules that we've been mentioning, NADH and FADH2, these pass electrons to the electron transport chain, and this is again located in the inner mitochondrial membrane. Now this in turn, these carriers pass them on to other proteins in the chain, and the energy available in the electrons is used ultimately to pump protons or hydrogen ions from the matrix of the mitochondrion across the inner mitochondrial membrane. Now this stores energy in the form of a transmembrane electrochemical gradient. Now as the protons move back across the inner membrane through the enzyme ATP synthase, that electrochemical gradient is used to power the direct production of ATP. So the movement of ions, in this case the protons, down their concentration gradient or their electrochemical gradient through that selectively permeable inner mitochondrial membrane, again, is called chemiosmosis. Now more specifically, it relates to the generation of ATP by the movement of hydrogen ions across the membrane during cellular respiration, and also, as we saw before, during photosynthesis. So the flow of protons back into the matrix of the mitochondrion via that ATP synthase molecule provides enough energy for ADP to combine with inorganic phosphate to form ATP. So let's look at a tally of where all these ATPs are coming from. So the total number, as we said before, of ATP molecules generated when one molecule of glucose is completely oxidized to carbon dioxide and water under conditions of aerobic respiration can now be calculated by totaling up all of the ATP produced by substrate level phosphorylation and also all of the ATP molecules being produced by oxidative phosphorylation and chemiosmosis. So glucose forms 2 triose phosphate molecules, which ultimately are converted into 2 pyruvic acid molecules, or pyruvate. This is glycolysis. As we said before, this step requires 2 ATP, whereas this step here generates 4 ATP, and therefore the net production of ATP via glycolysis alone is 2 ATP molecules per molecule of glucose. Now, in so doing, we have also generated 2 molecules of reduced NAD or NADH. Now, once we form our 2 pyruvate molecules, these will then break down to form 2 acetyl-CoA molecules during the link reaction. Now, each one of those acetyl-CoA molecules will then feed into one cycle of Krebs cycle, and as we saw before, each turn of Krebs cycle directly produces 1 ATP by substrate level phosphorylation. However, all of these processes produce these reduced hydrogen carriers like NADH. So the electron transport chain is going to make use of the reducing power of these molecules ultimately to generate ATP by oxidative phosphorylation and also by chemiosmosis. So the two NADH molecules, remember each NADH molecule when it enters electron transport chain ultimately generates 3 molecules of ATP, therefore 2 NADH molecules formed by glycolysis of 1 glucose molecule will generate 6 ATP via the electron transport chain. But in the link reaction we're also producing 1 NADH molecule for every molecule of acetyl-CoA we're producing. So each one of these also enters the electron transport chain, giving another 6 ATP. In Krebs cycle, each rotation of Krebs cycle, as we saw, produces 3 NADH molecules and 1 FADH2 molecule. So if you've got 3 NADH molecules, each producing 3 ATP, and 1 FADH2 molecule producing 2 ATP, but you've got that twice, then ultimately you're going to produce 9 ATP here, 2 ATP here, 9 ATP here, 2 ATP here. And so the net production of ATP for all of these processes is 38 molecules of ATP. So you really need to be very, very confident in your ability to explain where all of the ATP molecules are coming from. And I would suggest that you get familiar with this scheme here. So substrate level phosphorylation and oxidative phosphorylation are basically the main sources of ATP. Oxidative phosphorylation again is the process by which ATP is formed as electrons are transferred from NADH and FADH2 to molecular oxygen ultimately by a series of electron carriers. Now this occurs at the electron transport chain where the presence of molecular oxygen acts as the terminal acceptor of electrons, and as we saw before, if you don't have that molecular oxygen available, the electron transport chain essentially seizes up and ceases to operate. So the electron transport chain, as long as there is oxygen available, will form 34 ATP out of the total of 38 ATP molecules which are generated per molecule of glucose. So again, at various points in the respiratory pathway, ATP is generated without the need for molecular oxygen. So during glycolysis and Krebs cycle, there are certain reactions which generate sufficient energy to build ATP from ADP and phosphate directly. Now, these are the reactions that generate at least 30 kilojoules of energy per molecule of oxygen. So the formation of ATP utilizing the energy from such reactions again is called substrate level phosphorylation. So to remind you, we've got 2 triose phosphate forming 2 GP to form 2 pyruvate. This process requires 2 ATP, whereas this process requires 2 ATP, this process produces 2 ATP plus another 2 ATP. This process produces 2 ATP for every 2 turns of the cycle. So these are the processes which directly produce ATP by the conversion of one substrate into another, simply due to the chemical energy released in those processes directly powering that phosphorylation. Now, during oxidative phosphorylation, one molecule of glucose there are 6 ATP molecules generated by this process of substrate level phosphorylation. So to summarize, we can basically show the origins of all of the ATP per molecule of glucose in the various stages of aerobic respiration, and we can summarize that in terms of ATP used for the various stages versus the ATP produced from substrate level phosphorylation, and also the ATP produced by the electron transport chain due to the reducing power of the NADH molecules and the FADH2 molecules produced at these various phases. So glycolysis uses 2 molecules of ATP per molecule of glucose. The link reaction does not require ATP. The Krebs cycle does not require an input of ATP. So far we've used 2 molecules of ATP. Now, ATP from substrate level phosphorylation, per molecule of glucose that enters glycolysis, we produce 4 molecules of ATP by substrate level phosphorylation. The link reaction again produces no ATP by substrate level phosphorylation. The Krebs cycle per molecule of glucose. Remember, per molecule of glucose you get 2 turns of Krebs cycle. Each turn of Krebs cycle generates 1 molecule of ATP by substrate level phosphorylation, giving 2 molecules of ATP per molecule of glucose. So far we have a production of 6 molecules of ATP by substrate level phosphorylation per molecule of glucose. But also during the course of these processes we generate these hydrogen carriers which have reducing power, which can then be used in oxidative phosphorylation to produce ATP via the electron transport chain. So we produce 2 NADH molecules by glycolysis. We produce 2 molecules of NADH by the link reaction. We produce a total of 6 molecules of NADH produced by the Krebs cycle. So this gives us, once you convert these into the production of ATP, rather we have 10 molecules of NADH produced. Now in terms of FADH2, FADH2 is only produced by Krebs cycle, and you produce 1 molecule of FADH2 per turn of Krebs cycle. So since we get 2 turns of Krebs cycle, we've produced a total of 2 molecules of FADH2 per molecule of glucose. So in terms of the total ATP tally then, here we have 10 times 3, because each molecule of NADH under aerobic conditions, when it enters the electron transport chain, will produce 3 molecules of ATP. Each molecule of FADH2, which enters the Krebs cycle under aerobic conditions and enters the electron transport chain rather under aerobic conditions, will produce 2 molecules of ATP. So in terms of the total ATP tally, the 10 NADH molecules yield 30 ATP. The 2 FADH2 molecules yield 4 ATP, therefore we have 34 molecules of ATP produced by oxidative phosphorylation, plus the 4 molecules of ATP net produced by glycolysis, and Krebs cycle yields 2 ATP molecules by substrate level phosphorylation. Therefore 6 molecules of ATP are produced by substrate level phosphorylation. However, we used up 2 molecules of ATP during glycolysis, and so the net production of ATP by all of these processes is 38 molecules of ATP. So here we see an electron micrograph of mitochondria again. So these are the cristae, the infoldings of the inner mitochondrial membrane here, and this scale bar illustrates the scale that we're looking at. 50 nanometers is this length here on this micrograph. So we've talked about the biochemistry of respiration and the biochemistry of photosynthesis. We now need to look at the next section of the specification, which deals with the energy flow in ecosystems. So all organisms ultimately have to obtain a supply of energy and nutrients from their environment in order to survive. So energy flows through the community of an ecosystem in one direction generally, with primary producers harvesting energy through either photosynthesis or chemosynthesis. Now the majority of autotrophs, self-feeders, are photoautotrophs that harness the energy of the sun and pass some of that energy to consumers through feeding pathways. Now the energy contained within the producers and the consumers is ultimately passed to the decomposers, and these are the organisms, fungi, bacteria and so on, which are responsible for the recycling of nutrients back into the ecosystem. So there is essentially a one-way flow of energy through the biotic community and a cycling of nutrients between the biotic and the abiotic components of the ecosystem. So we start with the sun. The majority of autotrophs, being photoautotrophs, will then harness that energy to produce sugars by the process of photosynthesis. So this requires an input of water and various minerals, and will then produce an output as a waste product of photosynthesis of oxygen, some of which is taken up by respiration of the plant. But it will also lead to the production of sugars, which then can lead to the production of energy-rich foods. These energy-rich foods produced by the primary producers are then consumed by the primary consumers, the herbivores, which are then consumed by the carnivores, which can then be eaten by various other omnivores. Now ultimately all of these organisms are respiring, and so the sugars, the energy, the chemical energy contained in the sugars can then at least partially be transferred into useful energy as ATP. Now, there are also processes involved in the ecosystem, such as combustion, which then will then re-release, along with respiration, the carbon dioxide back into the atmosphere, but also the production of fossil fuels, coal and oil, from the slow decomposition and chemical change of the remains of organisms is also an important feature of this cycle of energy in the ecosystem. So the transfer of food energy between the organisms in the ecosystem, we can track this by constructing food chains, food webs, pyramids of numbers, pyramids of biomass, pyramids of energy, and energy flow diagrams. So a food chain is a linear series of feeding relationships in which there is a transfer of energy and nutrients from one organism to the next. So here is an example of such a food chain. So food chains will name the organisms at each link in the chain, and these give us a descriptive flowchart type diagram which describes the passage of energy and nutrients through these trophic levels of the food chain. So we start with the autotrophs, the primary producers. We go on to the primary consumers, which feed on the primary producers. We then go on to the secondary consumers, which feed on the primary consumers, and tertiary consumers that feed on the secondary consumers. So each step along the pathway is referred to as a trophic level. So an important concept here is the idea of biomass, and we need to relate this to the losses of energy at each one of these trophic levels. Now, the dry mass of living material at each stage in the food chain, or the so-called biomass, is always less than it was at the previous stage. Now, the biomass and the chemical energy contained within the biomass becomes reduced at each trophic level. Now, the reason for this is because some of the materials and the energy are always lost in the organism's waste materials, which go off into the environment. But also, respiration supplies all the energy needs for the living processes, including movement. Now, this energy is not available to the next trophic level because it's been used by the organism at that trophic level to power its movement. So, if we're looking at the energy flow for a pond, in this diagram, the units representing kilojoules hitting 1 meter squared of the ecosystem per year, the sunlight energy is 2.65 million units per meter squared of the ecosystem. Now, 156,000 units of that are actually intercepted and used by the waterweed. Of that, only 1,500 units of energy are then passed on to the primary consumers, the snails. Of that, only 154 units are passed from the snails to the fish, and so we see here that there is a drastic decrease of available energy passed on to the next trophic level at each trophic level. So only a small proportion of the available energy, as we saw, is transferred from one trophic level to the next. Now, this is estimated on average to be about 10%. Now, the reason for this, as we saw, is because much of the energy is lost as dissipated or wasted heat into the surroundings during the respiratory processes that occur within each organism in the chain. Now, it's this loss of energy at each stage which limits the length of food chains, or the number of trophic levels, and it's actually rare for there to be more than 4 clearly different trophic levels. So most food chains actually don't go beyond the tertiary consumer, because by the time you get to the tertiary consumer, the available energy is the available energy to be passed on is insufficient to power or to provide sufficient energy and nutrients to another trophic level above this one. So here's an example of a food chain with 5 trophic levels. This is relatively, about as far as it ever gets. So even in this example, the snakes may well eat the grasshoppers, which would suggest that there are not enough, that there are not 5 distinct trophic levels. So we start with the periwinkles, the primary producers, which are eaten by the grasshoppers, the primary consumers, which are eaten by the frogs, the secondary consumers, which are in turn eaten by the snakes, the tertiary consumers. Now, it is rare to get a quaternary consumer, but the hawks in this case can be thought of as the top carnivores. So within most ecosystems, organisms will feed upon a variety of resources, and for this reason, many food chains will link together and coexist to form food webs. Now, linkages between these linear food chains will then create complicated networks of connections, and these are what we call food webs. So let's look at an example of this. So we start with phytoplankton, and then zooplankton, small fish such as minnows, which are eaten by larger fish such as pikes, which can then be eaten by fish-eating birds such as ospreys. Let me just turn down the volume on this PowerPoint so it's not quite so deafening. Sorry about that. Okay, so food chains in fresh water can then interrelate with each other because one organism may well be involved in one food chain, but it has other sources of nutrition where it feeds on multiple different organisms from different food chains, and therefore these individual food chains can connect with each other into rather complicated food webs, as you can see here. So the food chains and food webs, these are qualitative descriptions of the feeding relationships between organisms, but as scientists, qualitative descriptions only have limited value. We need to be able to quantify these relationships, and of particular interest to ecologists are quantifying the numbers of organisms, the biomass of the organisms at each trophic level, and the energy available at each trophic level. So we're going to look at various different ways of conceptualizing this. So a different concept for representing feeding relationships is the pyramid of numbers, where the trophic levels are displayed as a series of steps of differing widths, like this. So the area of each step in the pyramid is proportional to the number of organisms present at each level. So you have a relatively large number of primary producers, a smaller number of primary consumers, a correspondingly smaller number of secondary consumers that feed on the primary consumers, and so on. So here's an example that we saw before. So the food chain that we saw before can be represented in terms of a pyramid of numbers here, because there are vast numbers of periwinkles that feed a relatively lower population of grasshoppers. Those grasshoppers will feed a smaller population of frogs, because each frog, to sustain itself, needs to eat multiple grasshoppers. In turn, each snake needs to eat multiple frogs, and so the number of organisms at this trophic level would be less, and the top carnivores will always have the fewest number of organisms at any trophic level. So here's another aquatic ecosystem represented as a pyramid of numbers. Now, in terms of large producers, so a single large producer can sometimes support many primary consumers. So for example, let me just go back one here. So you can have, for example, one plant which feeds a very large number of very small primary producers. And so in terms of a pyramid of numbers, this would be rather unusual, but it can happen. So you can have a rather smaller number of these large producers feeding a larger number of the trophic level above them, the primary consumers. Now, food chains involving parasitic relationships produce inverted pyramids of numbers. So for example, here we have sycamore trees, the primary producers. The sycamore trees, because they are large, are fed upon by larger numbers of primary consumers, caterpillars, because each sycamore tree can support many, many caterpillars. And so each caterpillar, though, is parasitized upon by parasitic wasps or larvae. So because each parasitic wasp there are larger numbers of the parasites than there are of the hosts, this is why we get this inverted pyramid of numbers in this parasitic case. So in this example, it's completely inverted. A large single producer supports many caterpillar, each caterpillar supports many parasites. So if we have more than one parasitic relationship, we can get rather strange looking pyramids of numbers like these. So we have large numbers of primary producers supporting a relatively smaller number of primary consumers, the cows. The cows support an even smaller number of humans, but then we have parasites which live on the humans, which are larger in numbers. So a rather large number of fleas can live on one rather unfortunate human, but in turn, the mites are actually parasites that live on these parasites. So we have a parasite of parasites here, and again, one flea can actually be host to multiple mites. So we see here a rather strange shape of pyramid of numbers where we have more than one parasitic relationship. So we've talked about pyramids of numbers. Let's have a look at the other kind of representations which are useful in terms of quantitative relationships, which are the pyramids of biomass. So pyramids of numbers represent difficulties for comparing different ecosystems and producers of different sizes, because as we saw before, occasionally you can get these inverted and even stranger looking pyramids. Now, a better representation of the trophic levels within ecosystems is to represent the organisms at each trophic level in terms of the total dry mass per square meter of area. So the dry mass of living material determined for any trophic level is referred to as the biomass. So a pyramid of biomass is a diagrammatic way of representing the total dry mass, again in grams per square meter of area, of all of the organisms in each trophic level at a particular time. Now, such numbers will take into account both the size of the organisms as well as the numbers of the organisms represented in the pyramid. So here we can see a pyramid of biomass for a rose tree. Again, this is, as we saw before, in a pyramid of numbers, this trophic level was actually smaller than the trophic level above it. But when we take into account the dry mass at each trophic level per square meter of area of the ecosystem, we see that it's more usefully represented as an actual pyramid shape. So the pyramid of biomass takes into account the size of the organisms, but again, it's only a snapshot of the amount of material present at a particular time, the so-called standing crop. So here we see the pyramid of numbers for a rose tree and contrast that with the pyramid of biomass for a rose tree. So this graph shows the biomass of producers and primary consumers in a marine ecosystem at different times of the year. So phytoplankton, which are the microscopic producers in this ecosystem, are eaten by zooplankton, tiny animals which are primary consumers, and we can see here the graph for the phytoplankton and the graph for the zooplankton. So in terms of the material flow through an ecosystem, one thing to think about would be, how does this graph portray a major weakness of pyramids of biomass? So let's have a look at the limitations of pyramids of biomass. So again, looking at this diagram, we have the phytoplankton production of dry mass per square meter of the phytoplankton represented by this red dotted line, and the value for the zooplankton represented by this black solid line. So it's clear from the graph that pyramids of biomass do not take into account the changes that occur over time, because at any given month, the relative proportions of these in terms of dry biomass per meter squared are changing. So phytoplankton are eaten by the zooplankton. So if we're exploring the limitations of biomass, it means that if samples are taken during spring, for example, then the phytoplankton populations are extremely large, and the pyramid of biomass assumes that the characteristic upright pyramid shape. But if we then measure the pyramid taken in May, of ocean samples taken in May, and this shows the biomass of the primary consumers may exceed that of the phytoplankton, whose temporarily low productivity due to those environmental conditions causes the pyramid of biomass to skew in this way. So the other thing is that pyramids of biomass also fail to take into account the fate of the primary production upon which the consumers depend. So some of the net production of producers is lost to the consumers as dead material, and food consumed by the consumers is not all converted into production. So consumers utilize some of their food for respiration, and also some undigested food is lost in the feces and therefore is not available to the next trophic level. So pyramids of biomass display the amount of material present at a trophic level at a given time, and these do not take into account the loss of material as it flows along the food chain. So we've seen that pyramid of numbers, pyramids of biomass, they can be useful in certain contexts, but they have these limitations. So sometimes we make use of a different kind of pyramid, a pyramid of energy. So the flow of materials along a food chain does not precisely reflect the flow of energy as we've seen. So a more accurate representation of the energetics of a food chain is obtained by measuring the energy content of the material and displaying this as a pyramid of energy, where the energy content of each trophic level is compared. So energy pyramids are constructed using energy values determined from a given area of the ecosystem over a specified period of time for each trophic level. Now this is usually measured in kilojoules per meter squared of the ecosystem per year, but it can be measured in other units as well. So the energy content of a material can be determined using calorimetry, which it basically involves the burning of a sample, a known mass of that material, under conditions where the energy released by that burning is used to heat a known mass of water such that the temperature change of that known mass of water can then be measured and then can be used to calculate the energy that was released for that mass of material burned. Now typically this is done in a device called a bomb calorimeter. Now this is a fairly standardized device which is used not just in a biological research context, but also for the measurement of the amount of energy contained within foods, for example. So burning the material in pure oxygen results in the complete combustion, and the energy released by that combustion is then used to heat a known volume of surrounding water. And so this change in the temperature of the water can then be used to calculate the energy release that was responsible for that change in temperature, because the specific heat capacity of the water is a known quantity. So this is basically the setup for a bomb calorimeter, and this is about as accurate as you can get for calorimetry, even though there are some losses to the environment here. So the potential energy contained in the sample is expressed in kilojoules, where 4.24 kilojoules is equivalent to the heat required to raise the temperature of 1 kilogram of water by 1 degree centigrade. So the specific heat capacity of water is 4,200 joules per kilogram of water per degree centigrade of increase in temperature. So again, pyramids of energy represent the flow of energy through each trophic level of an ecosystem over a specified period of time per meter squared of the ecosystem's area, and these are a measure of productivity. So energy pyramids display energy values typically in kilojoules per meter squared per year, and therefore they take into account the seasonal fluctuations in productivity. However, like the other pyramids, these also have their limitations. Now, these actually fail to display the fate of the energy at each trophic level, and they rarely include the energy flow to the decomposers and other parts of the ecosystem, and they cannot be used to determine the efficiency of a food chain. So here is one such pyramid of numbers here, and it's the one that we mentioned before. So let's look at energy flow diagrams. Now these illustrate the energy flow through communities. Now remember, the word community has a very specific definition in an ecological context. It is not the same thing as a population, so if you're at all unsure about that, then go ahead and ask in the Q&A box, and I'll be happy to explain the distinction. So energy flow diagrams, in contrast to pyramids of numbers, pyramids of biomass, energy pyramids, these include values for respiratory losses and energy flow to the decomposers. So information from energy flow diagrams can be used to calculate ecological efficiencies. So here is one such energy flow diagram. Now ecological efficiency is the net production of new biomass at each trophic level as a percentage of the total energy flowing through that trophic level. So therefore, for photoautotrophs, photosynthetic efficiency is determined like this. So photosynthetic industry for photoautotrophs as a percentage is equal to the net production divided by the light energy absorbed multiplied by 100% to get a percentage. So green plants only capture about 0.2% of the solar energy which actually reaches them, and then turn this into gross primary production. Now 50% of the sunlight passes straight through a leaf, and 5% is reflected from the surfaces of the leaf. Now of the sunlight that actually reaches the chloroplasts, only the red and blue regions of the spectrum are actually used, as we saw before. So the green light is reflected and lost, and this is again what gives the leaves their green colour. So photosynthesis actually overall is quite an inefficient process. So the majority of the incident light is not absorbed. Some of it is reflected, some of it is transmitted, and it's only those blue and red frequencies which are absorbed, and even those frequencies, not 100% of them are absorbed. So here's a problem that you guys can try for yourselves. Explain the meaning of gross primary production, explain the meaning of the term net primary production, and calculate the photosynthetic efficiency of the phytoplankton. Now, we've come to the end of the third hour of this webinar, and we have covered, I'd say, probably again about 2/3 of the available content of this topic. I'm happy to continue if those of you in the private Zoom room would like me to continue, but I will give you guys about a 5-minute break. In that 5 minutes, could you guys have an attempt at this problem, see if you can work out the answers to these questions, and also if you could all let me know. I see that the attrition rate has been relatively high, so we're down to about 13 students. So if those of you who are still in the Zoom room, could you just let me know if you'd prefer me to continue this at a later time, or if you'd be happy to hang around while we complete the coverage of these revision topics. If you could just type that into the chat, and then depending on what all of you guys decide, I will either continue and finish off the remainder of this topic, which will probably take another hour and a half. Alternatively, if you'd prefer, I can stop the webinar after the break and address your questions in the Q&A box, and then perhaps either today or tomorrow produce an additional video which I'll upload to the YouTube channel for the website to cover the remainder of this topic. So again, do let me know whichever you prefer. I'm happy to hang around, and obviously I will be hanging around to answer your questions that you've typed in the Q&A. But if you prefer me to continue this later on, I'm happy to do that as well. So I'll give you guys, say, a 5-minute break. So I will bring up that timer, and I will be back in 5 minutes time. So stretch your legs, grab yourself a cup of coffee, let me know if you'd like me to continue, and have a crack at that question, and I'll be back in 5 minutes. Okay everyone, we're back. Let me just see what the general consensus is here from the students in the private Zoom room here. Alrighty, so it looks like the majority of you are saying that you'd like me to stop the webinar here and follow up with another video to finish off the ideas that we covered in this webinar. I'm happy to do that. Alright, so I see, okay, cool. So yeah, let's just quickly finish off then by looking at the answers to that previous example, and then what I will do is I will shoot a additional video to cover the stuff that we didn't have time to cover here. I'll upload the video to the YouTube channel, but I will also upload the fully edited video containing both parts to your private coaching areas, those of you who are registered and enrolled on the Advanced Level Biology program of study on my website at Invisible College or invisible-college.com. Obviously, if you have any questions about the second half or rather the addendum video that I add to the end of this one, then of course you guys can get in touch with me via your private coaching areas, and I'll be happy to field and respond to any of those questions. So let's quickly finish off then by looking at that previous example here. Let me just bring this down a little bit. Okay, so let's have a look at the questions that I asked you guys. So the idea was to use the information from this energy flow diagram to calculate or to work out the following: to explain the meaning of the term gross primary production, explain the meaning of the term net primary production, and calculate the photosynthetic efficiency of the phytoplankton. So let me have a look at the chat here and see what you guys have come up with. Okay, it's looking pretty good. Well done, ladies and gents. Alright, so let's quickly work through that then. So the gross primary production, this is defined as the total energy fixed by photoautotrophs during photosynthesis. The net primary production, this is the energy stored as biomass, or the gross production minus the energy lost as heat in respiration. Now the photosynthetic efficiency you can calculate like this. You simply substitute the relevant values into that equation that we saw before. You evaluate, you get 2.15%. Okay, so I see that the vast majority of you got that correct. Well done. Alrighty, now I'm going to stop the YouTube live stream in just a moment and then start to address the Q&A box questions, which I see are rather numerous. But I see that, again, the general consensus is that you guys would like me to film an additional video and upload that to your private coaching areas to cover the stuff that we didn't get time to cover in this webinar. So referring back to the spec then, the only bits that we've got left to cover, so we're up to about here. We've got to cover a little bit more here and nutrient cycles, and so I will, so what I'll do is after I close out this webinar and I've answered all of your Q&A questions, I will then upload that probably this evening, and I will let you guys know via private message on the site and also via email, since many of you are signed up. Okay guys, so thank you for attending, and thank you to those of you who've watched on the YouTube live stream. I will edit the YouTube live stream video and then re-upload it as an edited video to the YouTube page. And once I have filmed that additional video to cover those outstanding ideas that we didn't get time to cover today, I'll upload that as part 2 to the YouTube website. Obviously the YouTube channel will not contain the Q&A session that I'm about to go into now. That is specifically for the benefit of those students who have booked tickets in advance and posed those questions. Alright everyone, I hope that was useful to you, and I will see you in the next webinar. Now the next webinar, let me just bring up the website here. The next webinar will be tomorrow, so let's go to the online lessons timetable. So the next webinar will be tomorrow at 11 AM and will be on the subject of control systems. So that will be all the stuff about homeostasis, hormonal control, the nervous system, and osmoregulation. That will be at 11 AM again. And again, the scheduled time is 3 hours. But again, if we do go over that, then I'll happily either continue, depending on what you guys want, or I will go ahead and film an additional video for you. So once again, I hope that was useful. Thank you for attending, and I will see you in the next webinar.